and everybody to the day 2 of our AIIMS ASM webinar. I welcome you all to the second day of our AIIMS ASM webinar, Antibiotic Resistance, a Renewed Fight. At the outset, we would like to start our program with the Vandana. I request the organizers to kindly play the video. Thank you so much, Dr. Saujanya, for the beautifully sung Saraswati Vandana. I would now like to invite Dr. Rama Chaudhary, the organizing chairperson, for starting the event.
Very good afternoon to all of you, and welcome to the second day of the webinar on antibiotic resistance renewed fight. I'm so happy that more than 400 delegates actually logged in yesterday, and we are expecting much more crowd today as well. As you were there yesterday, the first day was totally focusing on antibiotic resistance, where we had perspective of WHO by Dr. Anuj Sharma. Then we had CDC perspective by Dr. Daniel, followed by Indian perspective, Dr. Kamini Walia, and also approaches to One Health by Dr. Rajesh Bhatia. We also had wonderful talk by Dr. Arun Lok Chakravarti on antifungal resistance, which was followed by a panel discussion. And the discussion was so interesting. We kept on having various issues on antibiotic resistance being discussed, and mostly talking about how we can take antimicrobial stewardship to the remote places in India. And it was very well said that when we can take COVID testing to all the remote areas of India, then why not antimicrobial stewardship? And now the Today's webinar is totally going to focus on alternative therapies, and we have very distinguished speakers from all over the world. And I'm very happy that today our Dean of All India Institute of Medical Sciences, New Delhi, is here to address the audience. And we also have our Research Dean, Dr. Subrata Sinha, with us. And after Dr. Anita, I will request him to address the audience.
I request the organizers to please play the video of Dr. The Dean, ma'am, Dean Academics.
A very good morning to all the distinguished speakers who are present in this webinar from all around the world, and dear delegates. I'm very, very happy to learn that Dr. Rama Chaudhary, the head of microbiology at All India Institute of Medical Sciences, New Delhi, has organized a webinar on a very, very contemporary topic, that is antibiotic or antimicrobial resistance, which is really a big challenge in the current era as is being faced in the healthcare settings. I think antibiotics have been used very indiscriminately, and I think the medical fraternity is to be blamed to some extent. And this has really led on to the problem of antibiotic resistance. And current COVID pandemic has really made it worse because there has been a lot of antibiotic usage for patients, which has compounded the problem, or which is going to compound the problem of antibiotic resistance. The rates of hospital admissions have also increased tremendously because of the COVID pandemic. And again, most of the patients who are moderately, at least moderately sick, are being given antibiotics, although it happens to be viral infection. And we are expecting a lot of multi-drug resistant organisms in the current scenario. The use of biocidal agents, that is being used for environmental and personal disinfection, can also select out drug resistant isolates. So this webinar is very timely, organized by Dr. Rama Chaudhary on 7th and 8th October, has been done in collaboration with some of the very prominent organizations, both international and national. For example, World Health Organization, CDC, many other international organizations based in USA, and some of our Indian institutes like Postgraduate Institute of Chandigarh, Banaras Hindu University, and the Indian Association of Medical Microbiologists, and there are many others. I believe yesterday, that was day one, went off very well with very good talks by Dr. Anuj Sharma, who is a WHO expert, Dr. Daniel of CDC, and many others. And it included topics such as healthcare associated infections, antimicrobial resistance, antifungal resistance, etc. And there was an interactive panel discussion that happened at the end of the day. Today, that is the second day, also looks very, very exciting with talks from, again, a lot of experts. And today, I think the discussion is going to be mainly on alternate therapies, including bacteriophages, nanoparticles, probiotics, and novel antibacterial agents, etc. I believe that such programs are very essential, not only for the knowledge update, but also for increasing awareness amongst the healthcare personals, more so during this time of COVID pandemic. And I think the information which will be collected by the delegates and participants over these 2 days will go a long way for the healthcare workers to design infection prevention and control guidelines which are tailored to their individual institutions in the context of our country, India. Ultimately, the information gathered will assist in reducing the irrational use of antibiotics and other antimicrobials. I think this information must also reach public at large through print and other social media and some awareness campaign, because I believe that public participation is very necessary and the program can be really successful if public is involved in this very good cause. I must congratulate Dr. Rama Chaudhary and her team for organizing this webinar on antibiotic resistance, a renewed fight over these 2 days. And I wish her all the success and all the best for the grand success of the webinar. Thank you.
Thank you, ma'am. It was so wonderful to have you here and for your encouraging words. I'm also very happy to have Dr. Subrata Sinha with us. He is our research dean, and he has been actually very encouraging and inspirational figure in our institute, whether it is the field of biochemistry, biotechnology, laboratory medicine, and now with the COVID. I think the whole diagnosis of COVID has been actually initiated, coordinated by him and his team. And I'm very happy to have him with us today. And I request him to please encourage us with his words.
Okay. Good morning, good afternoon, and good evening, depending on wherever in the world you are. So, I am very pleased to be saying a few words in this AIIMS ASM webinar on antibiotic resistance, the renewed fight. Now, I'm sure there will be many people participating who are from the pre-antibiotic era, or at least from the era where antibiotics were not so commonly used. I remember my mother telling me about two of my uncles who actually died of what she used to call septic ho gaya after they cut themselves when they were shaving. So this is the world that we are going to actually enter if we do not tackle the problem of antibiotic resistance in an emergency. And as you all know, from WHO onwards, everybody has declared it an emergency. And this is actually today, this webinar is on molecular mechanisms and it is on alternative therapies. But I'm sure everyone is aware that to tackle antibiotic resistance would require a combination of behavioral change, habits, prescription habits, the ability, to clutch at the prop of the strongest or the most widely effective antibiotic at the first instance. So we are moving into an era of molecular mechanisms, drug discovery, new antibiotics. Now, in a way, it's an evolutionary fight. If you look at it as an evolutionary terms, the bacteria are evolving to keep themselves alive, and we want to discover drugs at a rate which is faster, or discover therapies which are faster, so that we can survive with dignity and preserve the gains of the lifespan that we have made over the century. So with this, a number of approaches are there. There are pharmacological interventions, non-pharmacological interventions. In fact, microbial ecology, the microbiome analysis has come in a big way in making a conducive microbial ecology for the human species to flourish. So with this, I'm sure there is no end to the spectrum for alternative therapies. I myself in our lab, we work on one of our research areas is on recombinant antibody technologies. And recently I've been co-author in a paper on anti-COVID human recombinant antibodies. And this is something recently we heard that, ICMR has actually sanctioned the use of purified horse serum for treating COVID. I mean, I hope it never comes to that for the microbes. And in order to do this and to give us this kind of success, webinars such as this one are critical to enhance knowledge so that with our intelligence and adaptability, we can adapt faster than the microbe. And by a multi-pronged attack, we can be successful in our effort. So with this, I congratulate Professor Rama Chaudhary and her team for this very well thought out, very well organized webinar with a galaxy of erudite, innovative, and inspirational speakers. And I wish the conference and webinar all success. Thank you very much.
Thank you so much, Dr. Subhrata Sinha. And we will start today's scientific session. And Dr. Bimal Das and Dr. Priyam will take over it. Can I have the CV slide of Dr. Bimal Das? So Dr. Bimal Das is professor of microbiology at All India Institute of Medical Sciences. Though primarily he's looking after bacteriology, but I can say he's a real combination of bacteriologist, virologist, immunologist, and actually being trained in CDC in virology, he has been able to take care of all the disciplines very well. He's also instrumental in getting the accreditation of AIIMS, our microbiology department. And today, I think the Department of Microbiology is one of the department at AIIMS which has got accreditation. And I think we are always like inspired. The very good work Dr. Bimal is doing in the field of respiratory pathogens, especially Streptococcus pneumoniae, Haemophilus, and many others, and also meningitis. Especially during the years back when there was meningitis outbreak, he was actually taking control of the whole scenario very beautifully. And I invite Dr. Bimal now to chair the scientific session, and Dr. Priyam, who is going to be the moderator. Can I also have the slide of Dr. Priyam, please? Dr. Priyam is an assistant professor. She has joined the department recently, and she has been working in special bacterial pathogens and in anaerobic laboratory. She's also having a lot of interest in the field of probiotics, and very enthusiastic, very active person. It is always good to have young people with you, which makes you feel younger and youthful all the time. So I invite both Dr. Bimal and Dr. Priyam for the next session. Thank you. So they will be taking over now.
Thank you, Dr. Amma, for the very eloquent introduction. I'm sure I don't deserve it. I'm sure you will agree with me that yesterday we had a wonderful session on the present problem and the solution of AMR in India as well as the world. But today, I guess we will have a more exciting discourse on the novel and innovative approaches to tackle this antimicrobial problem in India as well as in the world. Specifically talking about the nanoparticle approach as well as bacteriophage. Bacteriophage, you will today there will be several talks on the therapeutic approaches of the bacteriophage, and that's going to be really exciting because we have a number of experts from all corners of the world on this particular topic to talk about. And we'll also be dealt with by Dr. Amit Dinda, Professor Pathology, on the nano approaches of how to tackle antibiotic resistance. First, I would like to invite Dr. Gopal Nath. He is the former HOD and Professor of Microbiology at the Institute of Medical Sciences, BHU. His research interest mainly involves gut microbiome, pathogenesis of salmonella infections, bacteriophage therapy. He has more than 240 publications on this area. He has screened several synthetic and natural compounds as a antimicrobial with antimicrobial property. He has more than 200 publications as well as his 35 years of teaching experience. He has guided many PhD students as well as MD students in his field. His specialty interest is bacteriophage therapy, and he has been awarded several fellowship to the National Academy of Medical Sciences. He has been also awarded Professor U.C. Chaturvedi and P.N. Chitani orations. I would like to invite Dr. Gopal Nath, and thank you for joining this webinar today. Dr. Gopal Nath, please.
Thank you very much. Thank you. So it is being shared.
Yes,
Yes,
sir,
yes,
it is
yes.
visible.
Yeah, yeah. It says
Yes, sir, it is visible.
okay, okay. Thank you very much. All of you, the organizing committee, for such a event, fortunate to be here. So, whatever I have done in the past, I shall be showing. And you see, this is the bacteriophage therapy of chronic wounds, the wounds which are not healing for the more than 6 weeks with the conventional therapy. Then what we do is that we are isolating the bacteria because we have got the time to isolate the bacteria. Then we are preparing the customized bacteriophages, and they usually having the count of 9 to the 9, and then using on the surface. So I shall be showing before going to the slides. The definition of the chronic wound is the wound that does not heal by the this simple method within 3 months of the duration with the conventional therapy. And chronic wounds often remain the inflammatory stage for too long and may never heal or may take years. So there are so many causes of the chronic wound. So there are systemic causes and there may be the local causes. And if you see here, this is metabolic disorder disease like diabetes, systemic diseases like rheumatoid arthritis, and other form of the chronic diseases such as HIV infection, old age, malnutrition, alcohol, narcotic abuse, and so many medicines like steroid, estrogens, and vitamin K, also smoking. And the non-healing also depends on the size of the ulcer, age of the ulcer, location of the ulcer. If it is there is constant injury, mechanism is not allowed to heal, and there may be opacity of the local circulation, there may be necrosis, suppuration, edema, and exposed bone. But the local causes are very important because I shall be throwing the light on this issue. The study reports from WHO itself way back in 2005 that inappropriate treatment of the acute traumatic wounds was the most common cause of the chronic wounds. And there is Chinese study also shows that the principal etiology of the chronic ulceration is trauma or traumatic wounds, basically complicated by the infection, which is further complicated by the biofilm formation. The other reasons for the persistence of ulcers are supposed to be diabetic in 4.9% cases, venous ulcer 6.5% cases, and pressure ulcers in 9.2% cases. So this is biofilm. You can see, all of you know, and we have talked yesterday a lot about the antimicrobial resistance and the film. So I'm skipping those reasons, and I'm just telling you that the biofilm are leading to the slow penetration by the antibiotics, because antibiotics may fail to penetrate beyond the surface layer of the biofilm, this yellow area. Then there is green area. The resistance phenotype may develop during the therapy, and they are unable to be killed by the antibiotics because they are the resistant variety. And because of the altered environment, this pink one, the zone of nutrient depletion or waste product accumulation, antibiotic action may be antagonized. So I am showing you, for example, one case that 66-year-old male, diabetic, more than 6 years, and suffering from chronic wound for more than 6 years. He has undergone several rounds of conventional debridement, local antiseptics, local and systemic antibiotics, but without any fruitful result. Rather suffered amputation of 2 toes. So I'm just showing few of the pictures. You see this, we started, the patient reported to us in May. Then we isolated the bacteria from the wound site. It was Staph aureus. And then we started the therapy on May 2nd. And you can see that every weekly picture is there, and you can find out the gradual improvement here. So this is one. There are two wounds. One is on the lateral side here, and one is here. And then you can see this is 16th May, almost 15-day past. And then this is 22nd May. You can see some sign of healing is visible. And here this is 29th May. Quite good means picture is here. And then on 6th June, you can find that the small ulcer is still there, means almost healed. And then this is on 13th June. This is on 20th June. And this is on 26th June. So we followed for a month, maybe up to the August. You can see the gradual 8th July. This one here and this one also, there is complete healing. And then this is 16th July. And this is 22nd July. Again, this is, we were just following that whether there is any recurrence or not. So it was not there. And then this is on 6th August. And you can see there is complete healing and complete cure. So the second case is the younger brother of one of the plastic surgeon. He tried all his best and he did twice the skin grafting also, but it failed. So he was having chronic ulcer for 2.5 years. 2 efforts of skin grafting, but with failure. And this is the picture we started on December 2018. This was also having the infection by the Staph aureus. So with the, and you can see the wound is like this in a bad shape. And then we started the therapy, and the weekly these pictures were taken. You can see the improvement. This one, then here you can see the gradually the margins are coming here, and then again this has become smaller on 24th January, and this is 25th January. This is looking like this. On 28th January it is still smaller. On 29th January it has gone very small in size, and the last picture is in March. You can see there is complete healing. So this is the effect of the bacteriophage therapy. So these are the pictures of the head and tail. Everybody knows about this, of our own bacteriophages isolated here in our lab. And we know that the respective phases were, in this case, we applied on the surface, covered the surface for overnight with sterile gauze till the surface became sterile. And usually 6 to 8 applications were needed. The gauze piece soaked with the bacteriophage solution, and the consequence photographs were taken for assessment of the healing. So what we got is that the total number of the cases we studied, because they were all failed cases with the conventional therapy, and then we started our treatment with the customized phages. 37 of them improved, and it is more than 82%. And few cases where we were not successful at the time of taking this data. The later 3 cases could be cured. It took longer time, more than the 3 months. But there was one case with large surface area with little decrease in surface area. But it seems it may take many more months. One patient with colonization with different bacteria, because he was having very, very poor hygiene, in fact. And it was not meant, the instructions which were given by us. The patient was having venous ulcer, although surface became sterile, but there was no decrease in wound side. One patient undergone amputation due to osteomyelitic changes, and one patient was lost during the follow-up. So this is the picture. You can see that we divided the patient's population in 2 groups, diabetics and non-diabetic. In case of non-diabetic patients, the healing started a bit faster. You can see here. This started on the day 21, but while it showed the complete healing by the end of 2nd month. But at the end of 3 months, almost statistically comparable results were obtained. 63.2% here, 69% cases healed here at the end of this one. But if you add all these things, it goes to 82 or 83%. So we wanted to see that which bacteria was more difficult to treat. So in that case, in fact, we just watched for this. There is a Bates-Jensen wound assessment tool. So we followed, there are so many criteria. You have to measure the size of the wound, depth of the wound, and many other things I shall be showing you later on in the coming slides. And here I wanted to show you that this is the Klebsiella pneumoniae infected wound, which was most resistant and it took more time. And the on day 90, some of the wounds still remain to be closed. 1.1 or 1.5 in case of 2 in case mixed infection. So it has difficult treat for the staph aureus after Klebsiella pneumoniae. So here you can see we just look for the many blood parameters, what was happening with the bacteriophage therapy. So significant difference or improvement could be observed in case of hemoglobin. There was a bit betterment of the hemoglobin level after the therapy, and also in case of lymphocyte count, it rose from 14.905 to 19.629%. And this difference was statistically significant. And this is again based on the Bates-Jensen criteria. And you can see that the improvement could be seen. You can see this is size, this is depth, edge, undermining, necrotic tissue, and necrotic tissue amount. And then you can see from the second month and P were there was an improvement within a week, but total healing could be obtained only at the end of 90 days in majority of the cases. So in this case also, if you see the criteria of the exudate amount, the surrounding skin color, peripheral tissue edema, peripheral tissue induration, granulation tissue, and epithelialization. Again, you can see the improvement was visible, very significant within a week. So I'm showing few of the pictures which were the part of that study, and you can see from here day 1, day 8, and day 30, day 60, and day 75. We started from here and we ended here at the end of 90 days. The second case here is RP 65-year male diabetic. Again, we started from there, this wound, the really very bad, all the exudates and the pus and other things are debris up
Dr.
there.
Nath,
Yeah.
or your voice is not clear. I think the line is poor. Can we repeat the last few sentences?
Okay.
Not...
So am I audible now?
Yeah, it's better now. Thank you.
Yeah. So in this case, you see, we started the therapy on day 1, and at the end of 75 days, we got this picture. So this patient was died. This is the second case where we got the improvement from here, and then we are here at the end of 90 days. So you can see gradually by the 3 weeks, we are getting the healthy granulation tissue, and it's so if we go for the third case, this is again there is one. And then we got from day again the healthy granulation the increase in size and 60 like this, and day 90 complete from but after in the first week, after first week only, you can see there is quite a significant improvement. So this is another case. This is plantar aspect is really very difficult to treat because mechanical injury is all the time there. So, but even in this case also, you can find that the improvement from here, which is much better here. There is decrease in size, and here the size has gone down to this much on day 60, and complete healing on day 90. So another case, see a lot of flows are involved, and in a very bad shape, this wound is there. That was a challenge to us. We started the therapy, and we ended at the end of our display. Again, this case, again, the plantar aspect, the deep tissue and thick tissue is involved. And then this is infected. We isolated the bacteria, prepared this one and started therapy. And ultimately, we ended up here. Another case, so I'm skipping because it's taking a lot of time. So, what bacteria or bacteriophages are doing? They are known to produce or to be able to induce many enzymes and many other products which are just degrading the extracellular matrix present in the biofilm, because biofilm is the major issue in treating the chronic wounds. Along with the antibiotic resistant bacterial infection, and they are also able to infect the persister cells remaining dormant but reactivating when they become metabolically active. When the bacteria become metabolically active, they again start multiplying. Some cultured biofilm also seem able to support the replication of the bacteriophages than comparable planktonic system. So this is very significant point. It has been seen that because this is the natural thing, the bacteriophages are much more adapted or feel better with growing with the biofilm form of the bacteria rather than the planktonic system. So it is perhaps unsurprising that the bacteriophages, as the natural predator of the bacteria, have ability to target the common form of the bacterial life, that is the biofilm. So this may be the reason why the bacteriophages are acting better here in this case. This is the picture showing that how the bacteriophages are attacking. So integrity of the biofilm is disintegrated with the enzymes produced by the bacteriophages. There are so many depolymerases and many other enzymes. There are lytic molecules are also produced. They are killing the bacteria directly. And then the prophages, the progeny of the bacteriophage are diffusing inside. They are just destroying the matrix. If the biofilms are clear, after them, you will also heal. So once the biofilm are clear, and then we have seen that if we are using the bacteriophage for single bacteria, and even if there are mixed bacterial infection in the biofilm, so the mixed one is also getting cured if one bacteria is getting cured. So therefore, in conclusion, I can say that topical bacteriophage application is effective for the treatment of non-healing wound, irrespective of the drug resistance status of the infecting bacteria, because the bacteriophages never bother about the status of the bacterial resistance. Their receptor site is different. If this is a dynamic process, we have to isolate the bacteriophages and usually to have the customized phases. However, if there is acute situation, in that cases we should have the broad spectrum activity by the bacteriophages, and we can use the cocktail of those bacteriophages for killing. And when we are getting the customized specific bacteriophages for the infecting bacteria, then we can switch over to the specific therapy. So by this way, we have found that 90% of the chronic non-healing wounds are being healed or cured by the bacteriophage therapy. So in all these cases, we found no serious adverse effect and or even an adverse effect at all. So it is very safe to use, and we have used safely. We have recently concluded one more thesis using this one on the acute large size wound infection, and we have got that within 22 days there is the healing, and we have got the chances of the skin grafting, while conventional therapy takes 45 days with the secondary intention or tertiary intention healing. So with these words, I'm again thankful to Dr. Rama Chaudhary. She gave me this opportunity, and it's difference because we are waiting since long. Has stopped. Thank you very much.
So that we can cover it in time.
Yeah. That's why I was trying my best.
Yeah, the questions we will take up in the panel discussions.
Yeah, I know. Okay, thank you.
I would
You're not audible, Dr. Priyam.
extremely sorry. I would now like to invite Dr. Dinda for the next presentation. The organizers, could you please put up the CV slide of sir? He is the Professor and Chief of Renal and Transplant Pathology, AIIMS, New Delhi. His areas of research interest include experimental pathology with interdisciplinary research in immunopathology, tissue engineering, cancer biology, nanomedicine, and regulatory sciences. He has more than 30 years of teaching experience, has published more than 370 articles in index journals, edited 4 books, and has written 14 chapters in various books. He has also filed 12 patents. He is the assessor for NABL, GLP, and GCP systems. He is the secretary of Indian Society of Nanomedicine, the former president of Indian Society of Renal and Transplant Pathology. He is the vice president of the Electron Microscopy Society of India and the Society of Biomaterials and Artificial Organs of India. He is the member of executive committee in Global Coalition of Regulatory Science Research. He has also been the visiting professor of Albert Einstein Medical College of USA and the University of New South Wales and Deakin University in Australia. I now hand over the screen to Dr. Dinda for his presentation.
Thank you very much, Dr. Priyam. I would like to really thank Professor Rama Chaudhary and the organizers to giving me the opportunity to talk to this audience in 15 minutes. Obviously, this is a very important topic so far as the current generation of the new formulations which are being developed to fight the especially multi-drug resistance bacterial infection. Now, the example that how nanotechnology is helpful, I think you can realize that there are several nanotechnology interventions which is currently being used to fight the COVID-19 infection. With this small introduction, I think those of you who doesn't have the concept of the exactly what do you mean by the nanotechnology or nanoscale. So basically, this is a science or technology where we bring down any material to the nano size, that is the nanometer scale size. So here in this photograph, you can see that the basically the virus is around say 100 nanometer average size, RBC is 7.2 micron. So we are talking about the virus size of this particle. The moment you bring down the size of this material to nanoscale, then these material they acquire the new kind of physical, chemical, and biological properties. Say, for example, the classical example you can we usually give for the gold. Normally, the gold mass is the golden color metal, but if you bring down their size, it changes its color, and weight to surface ratio, it generates enormous reactive surface ratio. So these kind of unique properties, which actually we can develop by nanotize a material, we can use it for different kind of medical and healthcare application. And not only the healthcare application, this is the one of the thing we have shown that we have reduced the size of this gold nanoparticle to 2 nanometer, 5 nanometer, and 10 nanometer. And you can see the different color of these particle changes. So also there are other properties changes according to the size. Now, the use of the nanotechnology is now almost everywhere in the large number of industry and any domain you can see, from the environmental remediation, electronics, the metal, everywhere. And obviously, this is an enabling technology. where you want to develop a new functionality to handle a new kind of develop a new solution, then nanotechnology is really, really helpful for that. So it is for precision medicine also, it is playing a big role. You can target the different cells, cellular receptors, subcellular organelles, and also even metabolic activities of the cells also you can target by different kind of sensing mechanism incorporated into the nanosystem. So it is predicted that in the next decade, the technology, there will be a shift of this technology. So new discipline, new industries, they will come based on the application of nanotechnology. But anyway, I'm coming back to today's the topic. So what is the rationale of the nanopharmaceuticals or nanotherapeutics? Now, whenever we do a nano based drug delivery, so obviously the amount of drug which is needed for therapeutic purpose, if the amount is markedly can be reduced, because you can target these to your site of the either infection or site of the disease, in particularly cancer, and there obviously you can have a much better efficacy. That's why this is a much safer that way for the individual which is getting this kind of treatment, and efficacy level is also high. And so that's why it is good for the both the treating the physicians and as well as the patient, and also for the industry, the new novel formulation can be developed using nanotechnology. Now, the infectious disease, there is a huge challenge, because I think already it has been discussed the yesterday, and also today it will be discussed again. So I'm not going into the details, but the issue is that the new infections are also emerging, and where the immediately the therapeutic development is a challenge. And so particularly example is COVID-19, where nanotechnology has done a great help. And for antimicrobial resistance also, the nano interventions are playing a big role. And we hope that in near future, several of these the products and several of these technologies will come into the market. I'll give you a bird's eye view that how some of these things are being worked out, and also the clinical trial phase and in the product phase. Now, the scope of this nano intervention for infectious disease, that you can target the biomolecules and other high efficacious thing in the sites very specifically. And the other example is the vaccine development. Also nanotechnology is being used, which I will not be touching, but that's also a big area. And there's another interesting area which is coming up. We are targeting it as a metal biotics. That means the different kind of metals has got a huge the antimicrobial property, and this is the one which is being explored by a lot of workers across the world. Now, we have already shown, it's my older work, that you can target the macrophages by the nanoparticle. Here we have used gelatin nanoparticle. And these particles are really targeting the reticuloendothelial system, macrophages, and other macrophages. So intracellular infection particularly can be handled much better way with this application of technology. You can load any of these conventional drug into the system, and gelatin is a totally biocompatible system. So this kind of technology can be easily developed for clinical trial and subsequent use. Now, the next generation of nanosystems which we have developed is basically lipid-based nanosystem. Here we have used a more of a combination of chitosan and cholesterol system, and also surface modifications we have done by pluronics. So that the interesting component is that you can use this system for intravenous systemic the administration. Also, the same thing with the help of the pluronics, we can use it for oral therapy. So here we are developing using this technology for development of the oral amphotericin B therapy. But anyway, today my topic is AMRs, and I'm coming back to the AMR. So these are the size of the particles which we have developed. For the AMR, the most important thing or successful thing is the different kind of metal nanoparticle, and the forerunner is the silver nanoparticle, which is being used extensively by different kind of the application. And for COVID particularly, I think the copper nanoparticles are highly effective. But for AMR, I think I'll be talking about showing you the general principle that how these metal nanoparticle acts on these bacteria. If you see in this cartoon that they act at multiple sites in different mechanisms. So that's why for multi-drug resistance, it doesn't actually affect this efficacy of these metal biotics or metal-based therapy. And but the problem of in this area also we have developed 2 or 3 intervention. See, one of the major challenge of these metal, particularly silver nanoparticle, is that if you apply on the wounds and if it is rapidly absorbed into the system, then there is a chance of systemic toxicity. So what do we try to do? We actually encapsulate this silver nanoparticle within another polymer. So here we have used the collagen, and to make it much more less ROS generating system for the wound healing or rapidly of the wound healing, we have functionalized it with glutathione. And with this, we have shown that it is even much more efficacious than this silver sulfadiazine topical application. So similarly, nowadays the lot of these nanoparticle, particularly silver nanoparticle, is generated by the green synthesis. In the green synthesis, obviously you can use many plant extract. So we have used the green tea, and we have shown that you can generate very good the efficacious silver nanoparticle with the biomolecules which are good biomolecules which are present in the green tea, and you can make the film, and with a large wound you can cover, particularly for burn kind of application. This should be highly efficacious kind of treatment. And this technology has an advantage that silver nanoparticle can be retained in this biofilm itself, and gradually it degrades into that. So your absorption into the systemic circulation of the silver nanoparticle will be much less. So that means the nanotechnology applications can be innovative. According to the application, you can do whatever you want to apply. Now, in the range of metal, as I have told you, the titanium oxide, the copper oxide, the iron zinc oxide, and some people have used magnesium oxide, but the most effective one is the silver nanoparticles. But the major problem of any metal biotics or metal nanosystem we use is that it's excretion from the body. The problem is that the metal has a high chance of sequestration in vital organ, particularly the kidney, and where it can produce significant damage. So that's why this is very important that if the researchers or someone wants to develop this kind of metal intervention for the treating the different infection, and if it goes inside the body, then one has to be careful that to show that how it can be excreted. So we have shown that size dependency is very important. If you reduce the size of this nanoparticle below 10 nanometer, then its excretion in through the urine is very fast. And here we have shown that the majority of it is excreted within a couple of minutes, and then within 30 days, almost everything is excreted. This is done in the mouse study. Now, as the silver is one of the preferred mechanism for treat many of these multi-drug resistance infection, particularly the burn and other areas. The problem is that you have to really careful about its safety and efficacy, safety particularly, and environmental contamination. Now, though nanosilver is used extensively in different formulation, there are some of these warning I want to put into this gathering or audience, that you have to be careful, because more and more silver we use for different kind of wound intervention, there is a potential that a lot of silver can go into the sewages where it can affect your nitrifying bacteria, and that may have a bad effect to the environmental ecosystem. But anyway, still within safe limit, I think one can have this kind of the application development through nano intervention. The another huge area which is also coming up with the nano intervention is the nanodrug delivery system for antimicrobial peptide. I'm sure that you have already have listened, or this is a very important group of compound which is being tried. So more than 3,000 AMPs has been described, out of which the successful clinical trials are very few. The major problem is that low plasma stability, high susceptibility for the cleavage, and low therapeutic window for high chance of toxicity and immunoinflammatory effect. So these side effects, if you can reduce the side effect, and if we can use the targeted delivery of these AMP with the help of the nanosystem, then we can have a better efficacy. So liposome is one of the system which is being tried to have a specific delivery of these AMPs. and the different kind of lipid formulations. I have given one of the example of the lipid formulation which we have developed in our lab, but we are also developing the structured nanolipid particle, solid lipid nanoparticle, and other form of lipid nanoparticle. So these are will be very good. So I think this is my last slide. So if you see that what happened with the nano intervention, there is a rapid progress in targeted nanodrug delivery system, which will be very useful for treat the multi-drug resistant organism infection. Understanding of the biokinetics of the metal nanoparticle is very important because their systemic use is always fraught with the danger of its sequestration and toxicity. But the development of multifunctional nanoparticle is also becoming very important. We can develop nanoparticles which can be stimulated through electrical stimulus, magnetic stimulus, and they may disrupt the biofilm. So those kind of work already we have initiated, and there is a huge scope in that kind of the technology development. New generation nanoformulation development with approved antibiotic may be easier path of fighting AMR than new drug development. See, nano empower it some of these the antibiotic which is otherwise not effective against these multi-drug resistant organism. With nano intervention, you can make them highly effective. But more exploration is needed for understanding of the nanoparticle microbial interaction. Thank you very much for your kind attention.
Thank you, Dr. Dinda. It was a really very enlightening. We'll go to the next topic. Can you have the CV slide of Professor Martha? It is there.
Hello, can you hear me?
Yeah, good morning, Dr. Martha.
Good morning.
Good to see you. We're actually afraid that you may not be able to link up, but fortunately you are with us.
Yes, I'm sorry, I can't seem to change the background, but I'm here.
First, let me spend a few moments to introduce you.
Okay, great.
Yeah, Professor Martha is a professor in the University of Leicester. I guess that's the way you pronounce?
Yeah, perfect. Yeah.
She's a world-leading person in the bacteriophage, and her interest is mainly looking at the development as well as making it a product development as well as commercialization of treatment of bacteriophages in human and animal pathogens. I'm actually surprised that her interest in bacteriophages not only spans on human and animal, but it also extends to the silkworm. I was actually surprised silkworm as well as marine bacteriophages. That was very interesting because silkworm is the silk that is produced from the mocha silk that is found in Assam. I'm from that area, so I know about the mocha silk. I'm really happy to learn that you are working on the bacteriophages that affects the silkworm. Just coming back to the introduction, Dr. Martha has several patents, and these are specifically related to therapeutic phases. One specifically that looks at the Clostridium difficile, which is one of the major public health problems these days. She has also a fast-based diagnostic test for Lyme diseases, as well as Salmonella phages mix that can be used for the treatment of Salmonella infections. Dr. Martha also is the editor-in-chief of a newly launched journal called Fast Therapy Application and Research. She is also involved in multiple clinical trials that looks at the use of bacteriophages in the treatment of respiratory infection and diabetic foot ulcers. She has been awarded several funds by the government of UK as well as Thailand government to look at the development, design, formulate, and evaluation of different phases in the treatment of livestock infection. Dr. Martha, you are very welcome, and please go ahead your enlightening discourse on this area, please.
Okay, thank you. And thank you for the very kind introduction. Yes, I was very happy to work on the phages of with the silkworms. I was approached by a young Indian scientist who was upset that the silkworms were dying because they were under pressure from the they were weakened by the tea industry that weakens the immune system of the insects. So he's now he came to my lab for 1 year and set up his own program back in India. So I still maintain a nice friendship and collaboration with him. I don't know if we can see the slides. Can we see? How do I? Because I sent my slides over, or do I share my screen with them on here? What's the best thing to do to technically? I assumed that they would be shared from your end, but if not, I can
upload the slides. आपके पास हैं? आप अपलोड कर दो.
You just share the slides.
Should I share my screen, or will they come?
No, we are doing it.
Okay, perfect.
Okay.
Maybe chicken chili.
Ma'am, if you could share your slide from your end.
There we are. Yeah, that's good. I can see them there.
Can you see the slide from our end?
I saw them for a second. They flashed up there for a second.
I think let me share because
I think we are trying, but there is some problem here.
here you tell her to share.
You only share your slides.
That's it. They're sharing now. That's it. I can see them now. Okay. So today, I want to talk with you about the work that I've been doing on Clostridium difficile bacteriophages. So Clostridium difficile is a major problem globally, cause of infectious diarrhea, and it's very difficult to treat with antibiotics. So we desperately need novel ways to treat it. And as was mentioned in the kind introduction, you can see on my next slide that I have mainly worked on phages that target Clostridium difficile and guts, but I also have other projects where we're working on phages that target urinary tract infections, particularly multi-drug resistant E. coli and Klebsiella. And also in Leicester, we have a lot of we have a very big research base for respiratory diseases. So I've worked on phages that target other respiratory conditions, so Haemophilus, Pseudomonas, and strep pneumonia. So one of the things that's kind of I've done a lot of really in my research is trying to find phages for some of these organisms where phages are not used as much. So both Clostridium species and these more difficult respiratory infections. Okay, and I've done a little bit of work with TB phages, intracellular phages in general, and looking at Lyme disease. So these organisms, such as C. diff, they're very much in the shadows, and one of the reasons is it was actually very difficult to find phages for some organisms. So for some things that were talked about in the phage, the very interesting phage talk that we just heard, that was looking at wounds where often wounds are contaminated by staph, E. coli, Pseudomonas, and these are relatively easy to find phages for. But other things such as Clostridium difficile, I screened many different environments, and the most successful, you can see here on my slide, the most successful environment was actually looking in anaerobic salt marshes. So in a way, to find your phages, you often need to go to the environment that they're in. Now we can see here, this is what I did to find my collection of phages after screening for many years. I realized these are salt marshes. This is my son helping me sample here. So we got lots of anaerobic mud. We incubated them with the bacteria, and then we looked for signs of then we immediately sequenced the genomes of these phages to check that they didn't have anything in them that was undesirable. And after much screening, we came to a short list of, these are my top 7 bacteriophages for Clostridium difficile. We can see that there are different morphologies, there are different types of myoviruses, and also one cipavirus that's very effective. So these phages kill the relevant strains that circulate mainly in the UK. But in India, you have very similar strains that circulate. Now, this figure here is now showing you the a cluster analysis based on the relatedness of these bacteriophages. One of the problems you have when you isolate new phages is that you see huge amounts of diversity. And all I really want you to see from this is that these 6 phages look like, these are the 6 myoviruses that looked like they were quite similar. You can see if there's a strip that goes up, that all the genes are in common, you can see they have many, many genes that are not in common. So there's a huge amount of diversity to try and unravel. And one of the ways we do this is to at least try and work out when genes are expressed. So we can infect our bacteria with the phages, and we can see throughout a killing of the bacteria which genes are expressed. And at least we then know that these genes are important during the transcriptional takeover. So this is important because if we want to use phages therapeutically, it's helpful to try and understand a little bit about how they actually work. And one of the other exciting things with these C. diff phages is that we didn't at all know how they attached. So we want to be able to use phages that attach and work in different ways to minimize resistance, because if they all attach to the same thing, you can drive resistance. So you may have a new microbial, but it will become worthless quite soon. And can you see these little dots? Just look at here, and the bottom right picture. Can you see the phage with the little dots? So we finally worked out what the tail fibers were by expressing lots of different proteins that we thought were putative, and then doing raising antibodies and doing electron microscopy to identify which the protein was. And actually, it's a whole new structure. So we can see this is my protein crystallography apparatus. I'm very excited because we found this protein that looks like nothing else. You can see the work with new phages spans right from the ecology guddling around in the mud to doing quite detailed genome, RNA, and crystallographic studies to identify the mechanism of the way the phages work. So what we care about in this meeting, we care about, well, do they actually work? And I'm showing you here, you can see these phages work well. Can you see here's a C. diff culture? And this is it being cleared by the phages. And then if we look at this graph, we can see, and the next graph, we can see that some phages completely kill, other phages completely kill, other So we use this screening process to identify the more aggressive phages. And we ran them through animal models. So we ran them through hamster models and showed that they worked very well. And we identified a good combination of phages that worked well. And they removed significantly reduced the amount of colonization that Clostridium had in the guts of the hamsters. So this is a standard model for C. difficile. Now, the animal work is very time consuming and quite expensive. So we have developed many other models to study the efficacy of the phages. And as was also mentioned in the previous talk, phages are very good in biofilms. So we can see, can you see this inset here, the clearings? They show very nicely that the phages have been able to wipe the biofilm clear of bacteria. We also developed a gut epithelial cell model, and we showed that they work. The phages work well. We can grow the human epithelial cells and grow the Clostridium on top, and the phages again are good at clearing the bacteria in these well systems. So we have evidence from the animals, from the biofilm, from human cells, and then the next model that we set up was then using this wax moth. And actually, this is a nice Indian connection. It's because the Indian scientist that came to my lab, I told him to bring his moths, silk moths, and he couldn't. He said they wouldn't live. So I got this a standard model of infection using Galeria. So can you see these caterpillars? When they're healthy, they're yellow. When they're dead, they're black. And then you can see them in the process of dying. So we worked out how to use this as a really good model of infection, and it correlates really, really well to the animal data. We worked out how many bacteria we need to give the caterpillar a 60-hour window where we can study them. And we can see that actually if we add phages, before we add bacteria, so if we use the phages prophylactically, we can actually see that we can prevent the colonization altogether. So I think there are going to be many gut diseases where probably prophylactic use of phages will be very effective. And we can also combine them well with antibiotics. And if we're going to use them as a therapeutic, we have to just add them several times. So if we noticed in this insect model that prophylactic use was best of phages, but actually if we added the phages many times, we could also save the majority of the insect larvae. So this is a very good model of infection that we can use to test the phages. And the final model I wanted to just quickly mention to you was we used artificial gut models. So this is where you get a hold of fecal samples from different individuals of different ages to have a large representative microbiome. And then we spike that with Clostridium difficile and check that the phages will just kill the C. diff. So with phages, we have this advantage of having this lovely specificity. And we wanted to check here that the phages would work and they wouldn't get trapped on the wrong bacteria. And can you see the blue and the purple line on that graph? That shows you that whether the phages were added prophylactically or after the C. diff was established, all of the C. diff was removed. Sorry, if you just go back to that slide one for 1 second. I just wanted to point out that you could see the experimental setup there. Oh, sorry, it's gone right to the end. I don't know if you could go back a couple of slides. Essentially, I showed what I wanted to show you was one more, please. Yeah, that's it. So we showed this is the experimental setup there, and also we sequenced the whole of the microbiome of these cultures following phage addition, and we showed that the phages did not shift the balance of the significantly of the rest of the bacteria. So we have indeed got a really nice specific antimicrobial that will go in and just remove the C. difficile. Now, in the title of my talk, I said challenges. So C. difficile phages are not completely trivial. They're difficult. They're difficult to find. They're difficult to grow. So we've also been working on these aspects to show that they can to be able to ultimately manufacture them at scale. So we need to be able to grow them in a defined media, remove the toxins that are released when you culture them, because the endotoxins from C. diff are released. So we've shown that we can get rid of those toxins. We've also shown we can encapsulate them in a pH-sensitive polymer so that they can make it through the acidic stomach and be released into the colon. So these practical things need to be done as well when you're developing a phage product. And now the other major problem with these C. difficile phages is that they contain genes which we don't really want to have in there. They contain genes that are actually integrases, which we're in the process of removing. So that's, I think, was on the next slide, if you can show that. So there's different ways to do that, and we're using different techniques. Okay, so I think in the interest of time, we just wanted me to talk for 15 minutes. I've probably done that. So if you just show me the last slide, I'll conclude with what I've told you. So I've shown you that the C. diff phages were difficult to find. No, this is okay. Fine. Is just the next slide, please. So anyway, I can summarize probably without the next in the other direction. I can summarize probably without the prompt. Essentially, I've shown you that we've got a large collection of C. diff phages that we're now characterizing the fundamental of biology with. You can see because they're so novel, there's a lot to find out. But hopefully I've shown you that convincing data from the different systems that the phages have a lot of potential to remove C. diff and retain the rest of the microbiota. So with that, I will happily, I don't know if you have time for questions, but I'll finish my presentation there. Thank you for your attention. And this is to thank my lab members at the end.
So thank you, Dr. Martha. This was really wonderful. I've been working on C. difficile, and I know how difficult it is to grow organisms itself, and then to find phages is another difficult task. And I really congratulate you and your team for this wonderful work. And it was really fortunate to have you. And I think we will keep talking about this topic in months to come. And we'll be happy to work with you and learn from you. So I will request you to please be logged in. We have a panel discussion at the end of it. And there are some questions coming up for you as well. Okay, so lovely. Please be with us for some more time. Thank you so much. Very wonderful presentation. Thank you. I hand over to Dr. Bimal now. Thank you.
Thank you, Dr. Martha. Really nice and gave us a lot of insight specifically into the bacteriophage when you try to find the difficult phages from the Clostridium difficile. The whole journey was really wonderful. Thank you so much.
Pleasure.
Now I would like to invite Dr. Ramesh Nasimuthu. I hope I'm pronouncing your name correct. Dr. Ramesh has done his master's in applied microbiology and is currently working as an assistant professor in the School of Biosciences and Technology, that is SBST, Vellore Institute of Technology, Tamil Nadu. His areas of research interest include antimicrobial resistance and phage therapy. He has been awarded various prizes like the British Society of Antimicrobial Chemotherapy (BSAC) in 2009-10 by UK, the Young Scientist Fellow in DST SERB, and he has also received various travel grants. He was the Trichy and Vellore coordinator of Indian Initiative for Management of Antimicrobial Resistance (IIMAR) from 2009 to 2015, and was also the National Joint Coordinator in IIMAR. He is a member of Indian Initiative for Management of Antimicrobial Resistance, the ICMID Society, American Society for Microbiology, a member in EPICOR, the ProMed Mainland International Society for Infectious Diseases, Brookline, and many other Indian societies. I now request Dr. Ramesh to give his lucid presentation.
Great day to everyone. I hope my audio and PPT is on. Shall I continue?
Yes, sir.
Yeah, thank you. So I thank Dr. Rama Chaudhary for giving this excellent opportunity to join this. So I'm Dr. Ramesh, as he mentioned. Thanks for the profile, and also I'm working in the field of antibiotic resistance bacteriophage therapy. So I just started this phage work in 2008 because of I just we are seeing that the every day we are losing the available antibiotics. So therefore I decided okay, we should start the bacteriophages work. So today I'm just going to discuss about the therapeutical characterization of bacteriophages effective against the carbapenem 1 colistin resistant enterobacteriaceae. So the AMR and phage. So infections due to antibiotic resistant bacteria are increasing globally, and this needs immediate attention. Bacteriophages are considered an effective alternative for the treatment of bacterial infections, especially MDR, XDR, and PDR. Phage therapy is the therapeutic use of bacteriophages to treat highly drug-resistant bacterial infections. The current surge in bacteriophage therapy is motivated mainly because of the emergence of antibiotic-resistant bacteria in clinics. So in my lab, so what I don't want to add more point in AMR since we have a session on a. We have 2,000 isolates, so 1,500 gram negative and 500 gram positive bacterium from Tamil Nadu and Mizoram. So we do all genotypic and phenotypic of screening of MDR genes. So we focus on cephalosporin resistance, carbapenem, colistin, tricycline, MRSA, and MSSA. And so these are the genes, resistant genes we are screening in our laboratory. So far we have screened 79 CTXM, 38 NDM, 32 oxalate, 48 like, 12 IMP1, 1 DIM1, and 5 JES, and then 58 MECA, and then we have we sequence everything and then submit it to the GenBank, and then we do sickness to identify the strain, and then we have so far sequenced 4 integrant sequenced from the MDR pathogens. So this is some notable work regarding the AMR. So there are a few, as you all know that, the clinical pathogen gram negative. So the E. coli, Klebsiella pneumoniae, Klebsiella acetoca, Pseudomonas aeruginosa, Enterobacter cloacae. So they amplified the NDM1. So total number is 20. So some of the strains, E. coli, Kleb, and Klebsiella acetoca, they were amplified the OXA81. So in this result, we didn't got amplified in the MCR1 and 2, the colistin resistant. And for to notably integrants in the class 1, class 2, class 3, we got amplified, and it is the first report from the South India, and it is the it has the gene cassette, AMR gene cassette. That is very important here. So that line going to the phage therapy research. So the phage therapy, so first we start with the isolation of the samples, and then the plaque assay, and then you go for a host range assay, and then purification of the bacteriophages, and then the stability, and then stem. To do the morphological analysis, we go for a stem, and then one step go, and totally the life cycle, and then in vitro activity and DNA isolation, genome analysis. Then we are also trying with the phage cocktails and in vitro studies along with the wax moth larvae and zebrafish. It's all published. You can see the double agar overlay method. So you can see the clear plaque assays, lytic phages against the clinical pathogen. So here we used MDR, so multi-drug resistance. Even we are using PDR pathogens. Okay, because the available antibiotic density bacterium can be treated with the antibiotics, but we focus only on the phages should kill the MDR, PDR, and XDR. That is why we focus on it. So these are some of the images of the bacteriophages which we characterized and putted. So the first one is the HSF phage. It's a blank day, and then a Klebsiella phage belongs to the Podaviridae, and then the Enterobacter phage in the Myoviridae. Along with, we also trying with the Mycobacter phage, and then Serratia and Pseudomonas phage. These things are not published, and the above three are published. In our laboratory, we have bacteriophages for E. coli, Klebsiella pneumonia, Pseudomonas aeruginosa, Acinetobacter baumannii, Enterobacter cloacae, Proteus mirabilis, Serratia maricensis, Salmonella species, Staph aureus, and Citrobacter species, Enterobacter species, and Mycobacter sphygmatis. So we have this. We have the bacteriophages. So we used to have, keep only the the stable bacteriophages. We will not keep any stable bacteriophage, and it's not coming under our own category. Report, we have published 4 papers, notably in Class 1 BMC Scientific Reports and Frontiers in Microbiology. So the first paper, the completely characterized HRSF phage, it's named MyPSH1131. So including lyophilization and in vivo studies. So the lytic activity, it have killed 31 bacterium among 53 tested. So all resistant to carbapenem. Okay, and also it has a different pathotype. EPEC, EHEC, ETEC, EAEC, UPEC, and one unknown pathotype. So the absorption velocity. So when you talk about the absorption velocity of the bacteriophages, is 1.6 into 10 to the power of 9 per page, per cell, per ml in per minute. And the latency time was 20 minutes, and the burst size was 130 phage particles per infected cell. So the stability. So when we check with the stability, the pH range was 3 to 13. So the phage can be grown in the less pH of 3 and the highest of 13. So it is inactivated less than 3 and then greater than 13. So the temperature range 4 to 45 degrees Celsius. So we have now above the 45 degrees Celsius, the phages lost its viability. So the next study is the therapeutic characteristic of HRSF phage, MyPSH the 131, and then the Klebsiella phage, MyPSH 11235. So the Klebsiellas, the life cycle, the lytic activity was. No, it have killed 17 out of 44 different serotypes of Klebsiella pneumonia. So absorption velocity was 4.35 into 10 to the power of 9 phage per cell, ml, and minute. So latency time was 40 minutes, and the burst is 120 phage particles per infected cell. The pH range was 4 to 11. So it was inactivated above the 11 and below the 4 pH. The temperature range was 55 degrees Celsius. It can grow until, it can maintain until 55 degrees Celsius. And the Enterobacter phage, so this is called MyPSH1140. So the lytic activity, it killed 11 bacteria when tested with 15. So it can consist of 4 different species. Okay, and then the absorption velocity 2.8 into 10 to the power of 9 phage per cell per ml and minute. The latency time was 11 minutes. The burst size was 1. 11, and it was published in a well-known articles. Next. Yeah, good. So the other notable study from our laboratories, so lyophilization. So lyophilization played a major, because days we need to transport our bacteriophages to the throughout the global. So therefore, the phages should have a good stability. So far the result available in the current literature is saying that it's not pharmaceutically approved, and therefore we decided to make a excipients with a pharmaceutically approved, and we publish in scientific reports. So we have given we have used different the sugars is an excellent career protectant. Therefore we use different concentration, and finally the sucrose and gelatin gave a excellent stability to the bacteriophages, and also we have patented the live digestion of our method completely to the cadavers, okay, Ipavide, Mayavide, and Pordavide. So with the different excipients and also the different concentration. So the difference between our methods and the already available report is the we are making as a live flash, the powder can be directly mixing with the biological saline and then can be directly injected to the patients. No need to go for further development of the the phages and then purification and then extraction and then go for injection. We can directly go for a injection. So the sucrose and gelatin combination gave a excellent stability. So we also tried the new work with white Galleria melanella. So the Galleria melanella, a larger wax moth. So the study, already is published with the BMC microbiology. The in vivo studies of bacteriophages using Galleria melanella. So the Galleria melanella, a larger wax moth larvae, which is found to be a useful model to study the pathogenesis of and to study the antibacterial drugs. G. melanella is an ideal model. They do not require ethical level, have shorter lifespan, can be grown at 37 degrees Celsius, and they have similarities to vertebrates innate immune responses. So the use of Galleria melanella as an alternative model organism is gaining research interest. So the larval survivability studies were performed to evaluate the potential of phages against infection using Galleria melanella. So this is our experimental setup. So we have a 4 set. So set 1, set 2, set 3, and set 4. The set 1 control and saline, and the set 2 also control with bacteria alone, and the set 3 control phage alone. And the test we set 4 we further divide into 2. One is a single dose and then multiple dose. So the single dose, the infected with bacteria, the concentration of 10 to the power of 8 CFU per ml. The treated with a single dose of phages, so 10 to the power of 4 PFE per ml. The concentration same here, but the multiple dose we are given. 0th hour, 16th hour, 12th hour, and 24th hour. This is our experimental setup. Yeah, you can see here the Galleria melanella is live and healthy. Okay, worms, creamy white in color. So this is the dead worms. Okay, and dark spot. So when also we need to see the movement of the worm. So we got this worm from the Department of Entomology, the Karnataka Agricultural University. So we got it, and so based on this, the result, we can conclude that okay, yeah. So the control, so see the activity of HSF is single dose. So the control, so full of survivability. There is because the phage and saline did not inhibit the growth of kill the larvae. Bacteria alone, there is after 48 hours the pathogenic bacterium kills the worm. So when you see the treated with the E. coli phages, so you can see the first dose. The survivability is just 75% if you single dose. So you can see here after 96 hours the worm was killed. So because of still we need to give the concentration of phages and the dose should be increased.
Sir, only 2 minutes left.
Yeah. Of each E. coli, the multiple dose. So the multiple dose, when we increase to until the dose 2, it didn't kill, and when it increased, the level was surveyed, and also we have checked the bacteria which is present. Same for Klebsiella we did, and then for multiple dose for Klebsiella. So here 100 survivability, we need to go for the concentration. The dose should be increased to 3 and 4. But until 1 and 2, it survives. It's showing the enterobacterial phase. So the same here, and then multiple dose enterobacterial here. Okay, so the same all the category are bacteriophages, but only the in case of Klebsiella, it killed it within a single dose, but there is a no, the movement of worm is less when compared with the other phages. So we also did the cocktail studies. We mixed the bacteria and then infected. Sorry, and then we have seen that. So when we go for a cocktail studies, the concentration of the bacteriophage, the test, the concentration of doses should be increased. Okay, the first 0th hour, 6th hour, and 12th hour, and 24th hour. So finally, if you are increasing the dose, you can see here there is no bacterium here. So we are concluding that if you are using a cocktail study, if you have a multi-infections, the doses should be increased. So with this, I'm just stopping here. Since we also participating in antibiotic awareness work in India, so we are creating a lot of awareness program. This is the program we conducted, and yeah, with this, thank you so much. Thanks for your great time. Thank you all.
Thank you so much, sir. Thank you for your presentation. Now I request Dr. Bimal to introduce our next speaker, Dr. Taruna.
Dr. Ranjana.
Dr. Ranjana, sorry.
I would like to welcome Dr. Ranjana Pathania. Can you hear me, Dr. Ranjana?
Yes, hello.
Just a brief introduction of you. Dr.
Sure.
Ranjana Pathania, originally from Amritsar, the golden city of India. She has done her doctoral degree from IMTech CSI Chandigarh. Subsequently, she has completed postdoctoral fellowship at University of Massachusetts as well as McMaster University in Hamilton, Canada, and joined as a faculty assistant professor in the year 2007. And she is a full professor now. Her research interest is mainly mechanism of antibiotic resistance and development of novel strategies to counter determinant of drug resistance. Her approach has led to the understanding the role of non-coding RNAs that regulate antibiotic resistance in bacteria, and she has also identified other determinants, for example, novel efflux pumps, specifically in gram-negative bacteria like Acinetobacter. She also works with high throughput scanning methods for small molecule libraries. She targets antibiotic resistant gram-negative pathogen, and I think that's an excellent field of work. I would like to invite Dr. Ranjana Pathania to start the discourse today. Madam Ranjana Pathania, please.
Thank you, Professor Bimal, for your kind introduction. And before I start, I would like to thank Professor Rama Chaudhary as well as her team of organizers for giving me this opportunity to present my work here. So I hope the volume is good enough for everyone.
Yeah, it is okay.
Okay, great. So the title of my talk today is Antibacterial Potential of a Novel Nitrofuran IITR 6144 that we recently discovered at IIT Roorkee. Before the slides are, okay, they are moving now. Yes, so this slide is especially for undergrad and grad students, if we have any in the participants. And there are three things that I would like to point out when I talk about the timeline of antibiotic discovery. As you can see, as soon as any antibiotic was discovered, the things shown in blue here, as soon as an antibiotic was discovered, there was resistance against it. Also, there's a huge gap in discovery of antibiotics. The classes of antibiotics that were discovered, they mainly target gram positives, and there's no new antibiotic that has been discovered against gram negatives, as well as there's no new class of antibiotic discovered. So there's definitely a need for antibiotics which target novel pathways. The reason most of the antibiotics that have been discovered, they target gram positives, is because, as you can see here, the gram negative cell wall is really, really complex, less permeable, so the molecules cannot get inside. Also, the existing class of antibiotics that are used in the clinic, they target only 4 major pathways: cell wall synthesis, nucleic acid synthesis, protein synthesis, as well as membrane active agents. So there's definitely a need to explore new pathways, preferably multiple pathways as drug targets when we talk about new drugs. But this is again just a gentle reminder that when we take out infectious diseases, this is what a world without antibiotics would look like. No major surgeries, no hip or knee replacement, no cancer chemotherapy, no organ transplantation, no preterm infants. And again, for the students, antibiotic resistance is inevitable. It's not a matter of if, it's a matter of when, depending on which class of antibiotics we are misusing, abusing, or using too much. The global impact of antibiotic resistance, as all of you know, has been estimated to be 10 million deaths by 2050, costing us about 66 trillion pounds. So it's not just the resistance that's a problem. Even there's a separate category of bacterial dormant cells known as bacterial persisters, which can be a huge problem. So when we talk about discovering novel molecules, we need to think about these persisters, which are genotypically identical to the other cells, but then they can resist a lot of antibiotics, they can tolerate a lot of antibiotics, and they are responsible for causing recurring and hard to treat secondary infections that we usually see in the hospitals. So my group at IIT Roorkee, we follow a two-pronged approach against antibiotic resistance. One is we like to study the fundamental aspects of what are the mechanisms and how are they controlled in terms of small RNA regulators. On the applied aspects, we like to look for novel antibacterials using high throughput screening methodologies. We also like to look for adjunct molecules which can potentiate the ineffective antibiotics, and we're also working on antibiotic combinations. But today I'm going to talk about a novel antibacterial, a novel nitrofuran, which was recently discovered in micro. So a couple of years ago, we did a primary screening of a small molecule library, which had about 10,956 synthetic compounds, which had drug-like activity. And we screened these molecules at various levels using E. coli as our model system, because we wanted to look for molecules against gram-negative bacteria. And we ended up with 30 antibacterial lead molecules, which were then characterized for their antibacterial spectrum, for their in vitro pharmacodynamics, interaction with known antibiotics, molecular mechanism of action, as well as in vitro and in vivo toxicity. So today I'm going to talk about one such class, which is exemplified by IITR 6144. So we ended up isolating 4 analogs: 6144, 7711, 1324, and 6146. All of them had a nitrofuran moiety in their pharmacophore. And out of these, although we've synthesized about 40 odd analogs of these, characterized them all, but today I'm going to be talking about 6144 because it was the most potent of the analogs. And here's a list of antibiotics which are clinically used, which have the nitrofuran moiety: nitrofurantoin, furazolidone, all of them actually, all of them that are listed here. They are well-known antibiotics and are used in the clinic. So some of the data that I show you is going to be compared primarily with nitrofurantoin as well as furazolidone. So our first order of business was to look at the spectrum of lead series, and the data for 6144 is shown here. And as you can see, 6144 is compared with nitrofurantoin as well as furazolidone. And as you can see here, 6144 is effective against a bunch of gram-negative type strains, pathogens, as well as gram-positive type strains, as well as Mycobacterium species. It also inhibited a lot of clinical multi-drug resistant isolates, both in gram-negatives as well as gram-positives, as well as 6144 inhibited a bunch of anaerobic microorganisms. Anaerobic bacterial pathogens are also. So this screening against anaerobic bacterial pathogens was done in Professor Rama Chaudhary's lab. So from there, we went on to characterize its in vitro pharmacodynamic properties. first order of business was to see if 6144 was slowing down the growth or was it bactericidal. As you can see in the first graph here, this is bactericidal, and it exhibits enhanced post-antibiotic effect as compared to the 2 nitrofuran antibiotics used in the clinic. The post-antibiotic effect is doubled almost, and it showed no detectable resistance when we did frequency of resistance studies in the lab. Also, we tested this molecule against persister cells. We tested it against 2 kinds of persisters, stationary phase persisters as well as persisters isolated in response to various antibiotics. And as you can see, it inhibits both stationary phase as well as other kinds of persister cells, especially persisters of nitrofurans as well. So that was a good news. And then we went on and we studied the activity against against the biofilms, anti-biofilm activity. And as you can see from the first graph, it can, at various concentrations, 6144 can inhibit biofilms when it is added right from the beginning of formation of biofilms. And it can also disrupt, the lower one here, it can also disrupt preformed biofilms. And a comparison is done with nitrofuran as well as furazolidone in both the cases. So it had excellent anti-biofilm activity. So after this, we wanted to study its toxicity and do a safety analysis in vitro as well as in vivo. So for in vitro, we did hemolysis studies, the graph on the left, and we did toxicity studies against PBMCs, that is peripheral blood mononuclear cells. And as you can see, 6144 doesn't have any much hemolysis activity, as well as the cytotoxicity against PBMCs is not much, and it has a wider therapeutic index when we compare it to nitrofurantoin and furazolidone. Again, it was put into mice, and then we assessed various parameters, various serum parameters, and it was compared with these serum parameters in response to 6144 were compared with nitrofurantoin. And as you can see, most of the serum parameters, which are indicators of safety, are similar in both nitrofurantoin as well as 6144. I'm sorry, is someone trying to say something? No? Okay. So after toxicity studies, we went, sorry? AIIMS ASM is trying to speak, I think. Okay, I'm just going to continue. I hope it's nothing. Okay, so after toxicity studies, we wanted to assess its efficacy in animal models of infection. So we went ahead and we generated an animal model of Acinetobacter baumannii systemic infection. And after inducing the infection, we treated the animals with 6144, and then we harvested 4 organs, spleen, kidney, liver, and lungs, and we assessed their CFU counts. And what we found was, the graphs here show increasing concentrations of 6144 and comparison with nitrofurantoin. And as you can see, in all the organs, the CFU load has decreased considerably, showing that it exhibits excellent in vivo efficacy. And after this, we treated, we had a few probiotic strains, bacterial strains with us, and whatever strains we had about 5 or 6 probiotic strains, and they were treated with 6144. And as you can see the data here, 6144 doesn't inhibit any of the probiotic strains as compared to nitrofurantoin and furazolidone. So, so far, we thought 6144 displayed most features of, a theoretical nice antibiotic. But for us, the bigger question was, how does 6144 inhibit bacterial cells? What's the molecular mechanism of action? So we went back to literature, and what we saw was that nitrofurans have been listed as prodrugs. So in the cells, what nitrofurans do, on your left here, they enter the cells, and nitroreductases present in bacteria, they reduce these prodrugs into various intermediates which bind to multiple targets and then bring about the cell death. So we wanted to see if 6144 was a prodrug. So what we did was we took mutants, knockout mutants of nitroreductases. And as for other prodrugs of the nitrofuran class, MIC of 6144 increased twofold in the absence of nitroreductase NFSA, which probably hinted that 6144 was also a prodrug. To confirm this, what we did was we generated a resistant mutant over a period of 6 months by sequentially treating the cells with 6144 at sub-MIC concentration and then building up the concentration a little. And then we obtained this resistant mutant. The resistant mutant did show a growth defect, and then we sequence the genome of the resistant mutants in order to look at, in hope of finding some mutants which would, give us some idea about the mechanism of action. So to our surprise, we got an in-frame deletion in a gene termed RIBH, which is lumazine synthetase, which is an enzyme which is involved in synthesis of flavin mononucleotide, which is a cofactor for nitroreductases. So RIBH has been previously implicated in the literature for resistance to nitrofurantoin. So this confirmed that 6144 was a prodrug and was being modified inside the pathogen. Okay. Okay, our next job was to, treat the cells with 6144 and then view them under the microscope. So we saw the elongated cells, and these elongated cells, they were free of invaginated cell wall patterns. So then we asked the questions as to why would the cells filament. So there could be 3 reasons, either due to DNA damage, due to disruption of membrane potential, or due to inhibition of FTSZ. So nitrofuran derivatives are known to cause DNA damage in the literature. So our first question was, does 6144 cause DNA damage? So we did a tunnel assay, which is shown here on the left, and a DAPI imaging, and both of these techniques, they confirmed that 6144 was causing DNA damage, as well as the DNA damage repair deficient mutants were hypersensitive to 6144, indicating that it was causing DNA damage. We did not get any data for membrane potential, so 6144 was not causing any changes in membrane potential, but then we wanted to assess if it was inhibiting FTSZ ring, because if the FTSZ ring is inhibited, the cells are not going to separate. For this, we carried out an antisense to FTSZ and then looked for hypersensitivity to 6144. So plasmid control is on the left here. Another essential gene is on the right. And as you can see, the cells become hypersensitive to 6144, which indicate that FTSZ could be the probable target. This was confirmed by immunofluorescence microscopy. As you can see, the E. coli cells, which are untreated here, they have beautiful FTSZ ring. But when the cells are treated with 6144, something shown on the left happens, and the Z ring is diffuse, confirming that this was damaging FTSZ. Okay, so in the past, people have shown that bactericidal antibiotics, they cause ROS production, which we also saw in our case. But then we wanted to see whether this ROS production is cause or consequence of DNA damage. So what we did was we pre-treated the cells with vitamin C. And as you can see, the pre-treated cells, vitamin C is a quencher for ROS. The pre-treated cells, they still do not recover even when ROS levels are decreased. So this showed that ROS generation is a consequence and not the sole cause of cell death in 6144. So to conclude, we've shown that 6144 exhibits superior antibacterial properties in comparison to the existing nitrofuran antibiotics and can be developed further. So this is my working team, and I don't think I have time to acknowledge the funding agencies, so I'll just leave it at that. Thank you very much.
Thank you so much, ma'am, for your presentation of your beautiful work. I would now like to invite the next speaker of the day, Dr. Taruna Anand. Can you please put the CV slide of Dr. Taruna?
Yeah.
Dr. Taruna is working in ICAR, the National Research Center on AIIMS National Center for Veterinary Type Cultures, Sirsa Road, Hisar, Haryana. Her areas of research interest are bacteriophage biology. She has received various awards like the Indo-US Fellowship Award in 2018 by Department DST, availed at the Rockefeller University of USA. The Lal Bahadur Shastri Outstanding Young Scientist Award in 2017. The NAIP International Training in 2013 by Indian Council of Agricultural Research, availed in Germany. She has over 100 publications and has an H index of 12. She is a member of the Indian Virological Society and the Society for Bacteriophage Research and Therapy. Now, I request Dr. Taruna to kindly deliver your lecture on bacteriophage and AMR.
Yeah, thank you, Dr. Priyam, for such a nice introduction. Am I audible? Hello, am I audible to all?
Yes, ma'am.
So should I share the slides from my side, or you will be sharing them?
Ma'am, I think it will be more convenient for you to share your slides.
Yeah, sure. I'm sorry for this slide. Yeah, is it visible? Is it visible?
Yes, ma'am.
So, respected chair, co-chair, and respected seniors, dear participants and colleagues, this is a talk about.
Play your slides.
Is it fine? Changing?
Yes, ma'am.
Yes, ma'am.
Okay, so I'm continuing with it. The talk is about bacteriophages and AMR. Actually, in today's scenario of antimicrobial resistance, the bacteriophages are really gearing up, and we work with them since 8 years. And I'm working as a senior scientist and in charge bacteriophage lab at NRC Equines. So actually, this first slide is very important to show that antibiotics are everywhere around us. They are in our food, and be it animal food or plants, and they are present in our aquatic systems and through waters also. They are means we are only at the receiving end. And you would be really surprised to find that whatever antibiotics we eat, they are not absorbed in our body completely. Even the tetracyclines that we take, 70% of them are excreted as such, and all those antibiotics just go to the environment. This is a cross-country comparison of E. coli and Klebsiella pneumoniae resistance patterns, and the data for Center for Disease Dynamics, Economics and Policy. It shows that in 2015, India was having extreme number of Klebsiella pneumoniae and E. coli. And this was greater even in comparison to many other countries, even in comparison with China too. As you see, in past few decades, we have been witnessing that the number of antibiotics that are being discovered is very less, and those which are really getting patented, they are even more less. So in the past century, I could find this data that a very less number of antibiotics have been patented, the highest number being only 8 in the decade of 1950 to 1960. And 1980 onwards, we are hardly getting any new antibiotic that is getting patented. So it shows the intensity, like what is the number of, what is the resistance of the microbes, how they are dwelling around us. They are very smart, and they are emerging every day, and we are at the receiving end. As I deal with the animal sector, so I would like to here little bit let everybody know that in case of animals, the antibiotics are heavily used in animal farms, dairy farms, aquaculture, and many others. And they are used for the treatment of ill animals and for the control of spread of infections, prevention of illness in healthy animals during outbreaks, and as growth promoters also. And the sale of antibiotics has been immense. Even in different countries, means there is no country, hardly any country you will find where antibiotics would not be used in animals to increase their productivity. In dairy, fishes, piggery, means everywhere they are used. And so now this is a high time that we should say yes to alternatives of antibiotics. Now these alternatives may be antimicrobial peptides, they may be probiotics, they may be prebiotics, organic acids, essential oils, enzymes, and we are dealing with bacteriophages, and they are also known as green bullets because they are naturally occurring, and they haven't been found to cause any damage to the means environment in which they have been provided. Bacteriophages, this is a very general one. Everybody knows that they attach to the host with the help of receptors, and they insert their genomic material into the host, and the host then bursts because of the load of the progeny, and the progeny then spreads to other hosts to destroy them as well. Now, the potential applications of bacteriophages in veterinary sector, exclusively in dairy, aquaculture, and poultry, will be highlighted in this talk. The poultry is one of the most important source of development of multi-drug resistant bacteria. This is because antibiotics have been used here since ages. They have been used for as growth promoters and as feed additives. And the most common target pathogens here include Salmonella enteritidis, Salmonella typhimurium majorly, typhi, then Gallinarum, Salmonella pullorum, E. coli, then MRSA also is a big problem here, and Klebsiella pneumoniae. Apart from these, Campylobacter, Listeria, and Clostridium infections are very often encountered. And phages can be given under 3 heads here, means they can be applied in 3 forms. The first one is for therapy. That means they can be used for treatment of a disease. And they can also be used to reduce the load, bacterial load pre-slaughter. Then they can be used in biocontrol as biopreservative, as food decontaminating agents, and in packaging also. You will find many papers where phages are being incorporated into packaging materials. And they can be used as disinfectants on the surfaces and equipments, and for carcass disinfectant also. In case of dairy sector, the bacteriophages can be of immense help because they can be applied in 2 forms, 2 areas. First is if in case of therapy, for example, any animal is, for example, dairy animal, they mostly develop mastitis, and which causes very severe fever, and it leads to decreased milk yield, and it causes abnormality of the milk because the milk fouls and it has flakes, or it changes the color. Sometimes it becomes yellow, sometimes it becomes pink because of blood cells in it. And and there is a loss of appetite of the animal, and it is very difficult for the animal. This condition is very difficult sometimes. Sometimes these signs are not visible, but later on they become very much prominent as the bacteria keeps on spreading through rags, milking machines, and dirty lots and beddings. And though it is definitely a managemental problem, but sometimes it has been seen that those bacteria which generally they are not pathogenic to mastitis, to animals, when they reach from handlers' hands to the teats, they pose a problem for the animal. For example, Cocuria rhizophila, and there are some others also. Although the major target pathogens in mastitis are Staphylococcus, Streptococcus agalactiae, Streptococcus dysgalactiae, E. coli, Streptococcus oberis, Enterococcus faecalis, and Klebsiella pneumoniae, Klebsiella oxytoca. So this is the first area that you can directly give the phage for treatment of a disease. However, the product that is obtained from the animal, be it milk, be it like cheese, whatever is produced from using that milk, so there also the pathogens may dwell. And also the bacteriophages, there are reports that bacteriophages are being used. And here the major targets are again Salmonella, E. coli, Shigella, then in addition Yersinia, Bacillus, and Clostridium. So there are reports that phages against mastitis have been reported, and they have been, though I could see that they have been majorly used for in vitro studies. There is one study by Gill et al. 2006. This is the only study in large animals. However, they have shown that phage K that was tried at that time, it indicated a very limited efficacy. And a cocktail of 4 bacteriophages against E. coli was also reported. But adding all these reports all together, there has been lesser emphasis on in vivo trials, which is much required to arrive at a conclusive result. However, phages are available and have been tried in vitro for their effectivity against S. aureus and E. coli. Then in aquaculture also, they are being used. In Aeromonas, like there are infections of Aeromonas hydrophila and Aeromonas salmonicida. The fishes and loaches, trouts and salmons, they develop red furuncles on their body and they red patches on the body, and it decreases the acceptability of the product. Then in eels also Adversiella tarda. Then in catfishes there is a big problem of Flavobacterium, where the gills become gills become kind of they infected, and Streptococcus ini, which is in flounders, and Vibrio harvey in shrimp, that makes the fishes it means the shrimps bioluminescent, and their growth is also retarded. So in aquaculture also these bacteriophage applications are being tried, and they are have been overwhelming response for the bacteriophage application in aquaculture. Here you can find a table where phage therapy against bacterial pathogens in turkey, chicken, dogs, and even small animals like sheep, they have been tried, and they good results are being obtained, and these kind of results always prompt us to use bacteriophages. As we can see, they are amongst all of the alternatives of antimicrobials. These bacteriophages are the ones which have been studying from years, and we know them quite well. So, but the problem is that people are isolating them, but someday or later they lose it. So that is why nowadays the concept of bacteriophage banks is coming up. Ours is also a bacteriophage bank. So here the major steps that are involved in this bacteriophage bank maintaining is that first of all, we enrich the bacteriophages from raw samples, soil, water, sewage samples, and mostly people do like this only. And isolate the plaques, purify the plaques, and bulk culture them and preserve them. Then they keep on characterizing the bacteriophages by for morphology by transmission electron microscopy, then for temperature and pH sensitivity, growth characteristics, host range, and they try to develop suitable cocktails for a particular reason. So we need a bacteriophage bank actually for so that a panel of biologically active phages is available at a place and which may be screened against case-specific infection causing pathogens to select the most virulent or active phage against the provocative infectious bacteria. Now this needs a specialized diagnostic lab which is aware of different phage characteristics. A phage biologist prefers using lytic phages lacking any virulence, antibiotic resistance, or toxin genes. And this is lysogenic phages should never be used. The role of the phage repository becomes all the more important whenever we encounter a resistant bacteria. Then we can try many other phages that are available in the repository, and we can directly come to a conclusion that yes, we should pursue with this one or two phages further. Now the world has realized this thing, and many phage banks are coming up, including American Type Culture Collection in USA, then NBRC Japan, and Deutsche Center in Germany, the Netherlands Culture Collection Center, and China Center, and Wien, India, which is National Center for Veterinary Type Cultures. It is under Indian Council of Agricultural Research. Now, this is our profile, and we preserve bacteria, viruses, bacteriophages, recombinant clones, phage library, genomic DNA, and rumen microbes and dairy microbes also. In specific to bacteriophages, we have around 170 bacteriophages till now, which are against different genera and different strains, covering Staphylococcus, E. coli, then Acinetobacter, Salmonella, Klebsiella. So this is the basic characterization that every phage lab would like to do is the plaque characteristics and a spot test. And this is means we have to see the morphology of the bacteriophages, whether they are podoviridae or means myoviridae or cephuviridae. So there are different morphologies here, and we do test them for like what is the proteome and which is the major capsid protein encoding protein in gene encoding that protein in it. And the major activities that include are bacteriophage isolation, purification, and preservation. Now, bacteriophage isolation and purification is fine, but preservation is a bit tricky because generally we have seen that the different phages have got different requirements. Some would survive at one particular condition and some would definitely die at that condition. Then we do phage characterization. We provide on-demand supply of the phages, and we help people with training and knowledge sharing. But the major challenges that we face is checking and maintaining phage viability by revival at different intervals. So this is one of our study where we went for a phage therapy trial in mice against a virulent Klebsiella pneumoniae. And when we, in this mouse model, first the infection was established by nasal route, and then bacteriophage was given after 2 hours, and it was so helpful that it could completely help the bacteria to revive. As you see in the upper picture, the lungs are not so healthy. They are reddish, and there are red patches developed on the lungs. However, in the fast treated mice, the lungs are absolutely healthy. And yeah.
Please wrap up fast, ma'am. We are running short of time.
Yeah, please. Just give me 1 minute, okay? So the lungs were healthy and they were absolutely fine. And this phage was isolated from Ganga River. And this is a phage which was Aeromonas hydrophylla. It was published on the cover page of Journal of Basic Microbiology. And so our vision is that we need to enrich our repository with environmental and pathogenic phages against pathogenic bacteria. And we help the phage biologists with characterization at genomic and proteomic levels. And we are exploring phage therapy to combat bacterial infections, and we welcome all kinds of deposits. So we try to keep your phages safe. And this is an acknowledgement section. It is very essential that we should acknowledge all those who have been really associated with this work, and Dr. Nitin Virmani, Dr. B.C. Bera, Dr. Rajesh Vaid, and Dr. R.K. Singh, and Dr. Bendra Pathi. Dr. Bendra Pathi is DDG Animal Science now, and from the initiation days only, he has been associated with this. And I'm thankful for all these, and ICAR and Department of Biotechnology and DST for the timely fundings, what they have been providing. Thanks one and all.
Thank you so much,
for
ma'am.
this.
Now I hand over the mic to Dr. Vimal for introduction of our next speaker, Dr. Pinaki Panipuri. Please put the CV slide.
Dr. Panikrai, welcome to the webinar. Can you hear me? Can you hear me, Dr. Panikrai? Okay. Yes, sir. Dr. Panikrai is a professor of pediatrics at Georgetown University in Washington, D.C., and he is the director of international microbiome research. Initially, he has finished his medical graduation from MKCJ Medical College, Odisha in 1981. Subsequently, he completed his PhD from the University of Maryland, 1989. He has served as a Director of International Affairs at the President's Office at University of Maryland School. And he has many fundings from grants like NIH, National Institute of Health, Child Health Research, and Bill and Melinda Gates Foundation. He has many numerous publications that are published in Lancet, including Nature. But his main current interest is mainly microbiome. And as I already told you, he is the Director of International Microbiome Research. And fortunately for us, his association with All India Medical Sciences Department of Microbiology probably goes back about a decade or so, if I am right. Because I have seen him visiting the department for a long period of time. His seminal work in necrotizing enterocolitis and intestinal microbiota has won many awards, including Shannon Award from the NIH Director's Office, as well as Young Investigator Award from Eastern Society for Pediatric and Research Work. His current interest is role of discerning the role of infant gut microbiome as well as maternal vaginal microbiota in health and diseases, and addressing their role in stunting, preterm births, and other inflammatory conditions by mechanistic studies of immunomodulation. I welcome Dr. Pinaki Panigrai for this session of webinar. Please, Dr. Panigrai.
Could the organizers please play the video of Dr. Panigrai?
Good afternoon. Thank you, Dr. Chaudhary and the organizers for this opportunity. Since the symposium is coming to an end and I'm speaking to the choir, I don't have to do much convincing, which is a good thing. And antimicrobial resistance is everywhere. When I was in the medical school or a resident, this terminology was only heard in the context of ICU care. or maximum in the hospital that you acquire such resistance. But now it's all over the place. Our own gut, animal gut, the livestock, poultry, fish, pigs, environment, soil, and it's pretty serious affair. And it's so nice that you are holding this symposium in the middle of the pandemic, which is, I guess, equally important. And we shouldn't ignore these problems that we face today, and it's not going to go away. I will be speaking about probiotics. So before I do that, I will touch upon microbiome and how it develops and the disruption. Again, the culprit is or are antibiotics, and then how do we restore them? In which I will be talking about probiotics. I won't have time much on prebiotics or other botanicals that are also used for the same purpose. How does the human gut microbiome develop? I think most of you are familiar with it, that it starts very early at birth. And the microbiome comes from the mother or from the fomites or the environment around. And starting from the newborn period, if it gets damaged, disrupted, there are many deleterious effects. And in the newborn period, it is really bad. It can produce bad diseases like necrotizing enterocolitis, even sepsis. Equally concerning is if you are elderly or immunocompromised, the microbiome also plays a very important role. Probiotics it has been used in all organ systems, as you know today, and which I will again very quickly touch upon, and also tell you a little bit of the work that my research group has done. If time permits, I will go to the One Health Center at Asian Institute of Public Health University in Bhubaneswar. We can't really call the mammalian gut totally sterile. That's why I said almost sterile, because we know there is a microbiome in the placenta, although that has been questioned. But definitely the baby gets the microbiome from the mother's vaginal tract if it's a natural delivery, and then immediately from the environment and whosoever is taking care of the baby. What we know that there is a severe paucity of bacterial flora in the beginning, in the first days of life, and that is both in the number of species as well as quantity. And the change and quickly changes take place, especially when the baby is weaned from breast milk. And the gut first gets colonized with aerobic bacteria, and then that changes the redox potential, which allows microaerophilic organisms to come and settle. And finally, after several days, anaerobic species like strict anaerobes, like Bacteroides and Bifidobacteria, they settle in. What I want to emphasize here is all the good things that happen due to the exposure of the intestine to bacteria. They are very precise, and it has been shown that it is not due to age, just the postnatal age, it would have happened. And we all in the microbiome field, all of us know that there are, for each eukaryotic cell in the body, we have 10 bacterial cells, and they must be doing something good to us. This was about 20 years ago from Laura Hooper's lab, where they showed that if you have germ-free animals, the vascular, the intestinal perfusion doesn't happen or happens suboptimally. In a conventional animal in panel B, by 10th day, you can see that it is very well developed, nicely perfused. If you give on top of it some Bacteroides like Theta, Eta, Micron, then it's even better, which shows that indeed bacteria are needed, and lack of which can be deleterious. And so we will be slowly going towards the beneficial effect of those bacteria, and culminating into probiotics or defined good bacteria that we'll be talking about today. We just heard about acquisition of normal flora. How about drug-resistant bacteria, which is the topic of today? There have been several studies in different countries done, but I will just quickly show you what we did 10 years ago. This was a multi-center study, NIH funded, and Dr. Chaudhary was heading the labs at 4 different centers in different parts of India. It was surveillance of neonatal infection in the first 2 months of life, to 60 days of life. And what we found is really alarming, and this was 10 years ago. In Orissa, in the community site where the babies were born at home, the ESBL containing gram negatives, there were 1/3 of them had it in their stool. In the hospital in Bhubaneswar and in Rourkela, close to half, more than 40% had ESBL. In the tertiary care in Nair Hospital in Mumbai, more than 90% had ESBL, which was not that surprising, but it was pretty high that early in life. But for a baby that was born in the community at home, who has never seen a hospital, has never received antibiotics, to have 1/3 of them to have ESBL in their stool is quite alarming, because they're getting it either from their mother or from their environment, and it's not a trivial matter. So coming back to the human gut microbiome, what happens if it is disrupted? How to restore it, because that's what we are talking today. This is a nice picture where it shows in the middle that there is a normal intestine, we have gut microbiota, there is homeostasis, everything is good. On one hand, on one side, there is abuse of antimicrobial agents in animals, in the environment, water, air, soil, in the hospital, everywhere. And then that causes disruption to the mucus layer, and the bacterial translocation takes place, and final inflammation and disease, which can be blocked or reduced by multiple things that are shown here on the right-hand side, starting with phage therapy that you have already discussed, then probiotics, prebiotics, and the ultimate, which is fecal microbiota transplantation, or FMT, and that has been shown to do quite a bit of have good effect on C. difficile, and starting to be tested in other diseases. If you think about antimicrobial resistance, the first thing that comes to mind is C. diff disease, colitis and diarrhea. In refractory cases where antibiotics don't work, and it is pretty debilitating for the individual, now people are doing FMT, but probiotics have been tried in many papers, have been done, research studies, and a recent meta-analysis analysis of about 1,000 participants show that there is modest evidence to use probiotics in this particular disease. There are many other intestinal conditions where drug-resistant infection is a problem, like starting with diarrhea to other conditions. And there's I don't want to list all of them, but there are quite a few. And in each one of them, lactobacillus treatment works. Sometimes it works beautifully, sometimes it doesn't, which may be related to the strain or the species or the amount of lactobacillus being used, but there is enough evidence that it works. What's more interesting is the role of probiotics on refractory skin infections. I had seen a paper I tried and I couldn't find it. It was in Science. The title was something like killing fire with fire or something in those lines, where they had shown that how lactobacillus, I think, lactobacillus patches can treat ulcers that were not going away with traditional treatment with antibiotics. And there are many more papers available now, and we know that if they're drug-resistant skin infection also, probiotics can work. You will have these slides, so you can go over some of those nice important papers to see which one works and which one doesn't, but in general they do. I will tell you very quickly about a large randomized control trial that we did where we used symbiotics. Symbiotic is nothing but a combination of probiotic and a prebiotic. This was in an attempt to prevent sepsis in newborn infants in the first 60 days of life in India. And it was probably the largest study. We enrolled over 4,500 babies, and in the community setting, in 2 districts of Orissa, which were 250 miles apart, and we showed that there was significant reduction in sepsis as well as respiratory tract, low respiratory tract infection. Why I'm showing this to you is in this study, after it was not in the specific aims of the study, but Dr. P.R. Misra, who was handling cases around Bhubaneswar, he had collected stool samples for about up to a year, and then he examined the E. coli and Klebsiella that he had from those. And this has not been published. This is what he found is that in the first 2 months, first sample, the 60 days, it was plus or minus a few days was given. In that time, one could see that there was a significant reduction in the number of ESBL containing bacteria in stool in the probiotic treated arm. By the time the baby was 5 months old, 5 to 6 months old, or 9 to 12 months old, those effects kind of got washed away, and by 9th month there was no effect at all. This is probably because of the fact that the probiotics were given for 1 week after birth, so the impact may not be there, or other factors, and the microbiome might have changed. So here again we have a very strong evidence that indeed probiotic treated infants in this particular study showed that they somehow inhibited the acquisition of ESBL containing gram negatives. These are all, while talking about diseases or even animals or aquaculture, shrimp, birds, poultry, these are all living beings. But as we know, antibiotic resistance can be a problem and is a bigger or equally big problem in the hospital setting. So what do we do about that? In fact, some very bold studies have recently been done where people have gone ahead and sprayed the rooms and the patient rooms and the bathrooms with probiotics, with 3 different kinds of bacillus spores, because they will survive and easy to grow. What they found is quite interesting. They used 3 different kinds of bacillus, Subtilis, Pumilus, and Megatherium, and you can see the percentage of growth inhibition on those surfaces was quite interesting, and up to 97% in some. When they did the CFU for all different types of bacterial species, there was a significant reduction. Same was true for antibiotic resistance genes. in the treated one from 40-50% to about 100% reduction. This Italian group became a little bit more bold, or how do I say that, because they have already had seen the effect of probiotic spraying, they thought, let's see what will happen if we add phage, aerosolized phage spraying in the environment in the same rooms. So as you can see, first they had this PCHS, they call probiotic cleaning hygiene system. On top of that, they had spraying by nebulizing phage, and what they showed that this had additive effect, where there was about 100%, 90-70% better effect than just probiotics alone. At this point, one would question that yes, it is probiotics are working, whether in living beings or on inanimate objects. So what is the mechanism of action? Kunishima and colleagues, they have elegantly asked very pertinent questions that all of us that come to anybody's mind. And they have looked into growth and gene transcription, transmissibility, enzyme expression, and they have shown that indeed taking taking supernatant of Clostridium and Ephesium and L. plantarum in a dose-dependent manner, that indeed the growth was reduced. What was really unique and again interesting was that B. fragilis and even Bifidobacterium longum, they had no inhibitory effect whatsoever, which shows that there is something going on with specific bacteria and the factors that they release into the supernatant when they are cultured that are imparting the impact on these. Again, another strain that could suppress the expression of the CTX gene. And looking at all these, we can conclude that probiotics can probably be used to reduce or eliminate MDR infection at multiple body sites when it comes to a living being. It can also be used in the hospital setting, which is the hotbed, which is the reservoir of all these organisms carrying MDR. They can also benefit probiotics.
It's not live.
And phage or combination of those type of intervention. It is not clear whether it is one size will fit all, because they are using combination strains. They find good effect in some, not so good effect in other. So there is more research needs to be done before one could say that, okay, this is the right probiotic which is going to work against everything. There may be different strains needed, the way they work for surfaces versus intestine versus skin. But there is more than enough evidence, I would say compelling evidence, that one could move away from using antibiotics, whether it is in humans or breeding animals, poultry. The probiotics treatment has a lot of promise, and people are moving towards that. Even in the aquaculture, sometimes many countries don't allow antibiotics in shrimp. So the hatcheries are forced to use probiotics to suppress cholera and other Vibrio cholera and other infections that kill shrimp population, and also similar things in salmon and other aquaculture environments. And we all know what happens with poultry, which is really bad from this angle. and they are being given different types of probiotics now instead of antibiotics, or at least farmers are trying to reduce antibiotics as much as possible and see what happens with to manage the birds with probiotics. It's all good, kind of glamorous, but what about probiotics bacteria themselves? Do they carry antimicrobial resistance? And the answer is yes. In fact, they can carry it. Some of them carry plasmids, some can be chromosomally encoded. And once you introduce that bacteria, thinking that it does good things, it can
Sorry, we had to interrupt Dr. Panigrahi's video. We are actually running short of time. Now I would like to invite our last speaker for today, Dr. Yogesh. He is a professor of microbiology and the director of molecular laboratory at the SS Institute of Medical Sciences and Research Center, Karnataka. He has done his master's from Manipal University and has done 2 PhDs, one from Gulbarga University and the other one from St. John's Medical College, Karnataka. His areas of research interest include surveillance of antibiotic resistance, phage biology, pharmacokinetics and dynamics of bacteriophages, animal models to study the impact of bacteriophages as an antibacterial agent, and dengue epidemiology. With over 20 years of teaching and research experience, he has more than 100 publications. He is the vice president of the Indian Society of Bacteriophages and editor of 2 journals and has guided 3 PhD of 3 students. I welcome you, sir, for your talk on chimeric bacteriophages.
Thank you. Thank you very much.
The other bacteria that are around.
Could I share my slides?
Either in the
Yes, sir.
Several speakers speaking on strategies for AMR and alternative. Not much left, to tell, because from the yesterday I've been deleting a lot of slides. So I'll be just focusing a very few slides on my area of research.
So please put it on full screen. The slide share, the arrow button.
Yeah.
But I think there are network issues at your end. Your voice is actually breaking. Can't hear anything.
Hello.
Yes, sir.
Shilpa?
Sir, actually, there are network issues at your end. Dr. Vinod, sir, your voice is actually breaking a lot. Sir, if you could just tell about your research.
Is listening?
I think Dr. Vinod not logging has left probably because of connectivity issues. Sir, are you there?
No, ma'am. No, ma'am.
Okay, so I think we'll start the panel discussion and get back to him when he is there. Okay.
So I think we'll start with the panel discussion, and as soon as sir is able to log in, we will get back to him. I now invite Dr. Rama Chaudhary. She is the organizer of one of the organizers of the event, the main one. So, ma'am, please take the show for the panel discussion of today. So, can you please put the CV slides?
We will have the panel discussion now. Dr. Vinod is having some presentation problem right now. So I will just introduce some of the panelists. We have already, Dr. Vinod's slide was already projected. We also have Dr. Sharmishtha Day. Can we have Dr. Sharmishtha's slide, please? Dr. Sharmishtha is additional professor in the Department of Biophysics, and her areas of research interest are natural proteins, peptides of biological significance, biomarkers of cancer, biomarkers of aging, and age-related diseases. She has developed protein markers for oral cancer, pancreatic cancer, Parkinson's disease, Alzheimer's disease. She has also evaluated selected Ayurvedic plant products for their possible role in age-related diseases. She has also made some proteins which she has checked as an antimicrobial peptides. She has received AIIMS Excellence Award in 2012, 14, 15, 16, 17, and 18. In fact, every year, Dr. Day is getting an award for her research work. She has also received Surinder Marwa Award 2016 from ICMR and published more than 150 international journals. We also we have already introduced Dr. Martha. We have Dr. Sunita Grover with us. Thank you. So next one, Dr. Martha already shared. So Dr. Sunita is a former principal scientist and head in charge of molecular biology unit, and she's also former head dairy microbiology division NTRI. She's a fellow of National Academy of Agricultural Sciences, and she has been conferred with previous Young National Scientist Award. She's also authored textbook on comprehensive dairy microbiology and written ebooks on dairy biotechnology. Dr. Vinod, are you here now? So, I will just request you to put yourself on mute. And we have already introduced others. Dr. Kumar will also be joining in the panel discussion. So I will be starting with the questions now. So can you show the videos of the panelists? They can be put on now. The first question I want to address is to Dr. Sharmishtha Day. Is she there? Sharmishta, are you here?
Yes, ma'am, I'm here.
Yeah, so I would like you to share your experience. I request others to please mute yourself so that everyone can hear. What is your experience of using combinatorial drugs, like using antibiotics and peptide molecules together, and can we use them to overcome AMR?
So, ma'am, antimicrobial resistance is one of the biggest public health challenge. And this, I think this antimicrobial resistance developed due to the overdoses of the antibiotic. Can you hear me?
Yes, yes, we can hear you.
Yeah, so for the overdoses and for using a long period of time, so it can be overcome by using a combinatorial approach by using antibiotic with the along with the antimicrobial peptide, and this because these antimicrobial peptides are the uh, uh, these are very rapid onset of killing, broad spectrum of activity, and they do not easily select antibiotic resistance variant. And as they are killing very rapidly, they do not allow to form an resistance, and it shows a synergy with the classical antibiotic. The synergy is very main objective for the combinatorial therapy, which increase the activity and diminish the toxicity of the host. So it requires a less dose of antibiotic, and it kills rapidly with the combination. So it can overcome for developing the resistance by the microbes. So I can show two slides. My slides are not there. Ma'am, my slides. So I have developed two peptide. I cannot show the slide. Anyway, I can say. Where are the slides?
Can you share from your side?
It's not accessible. I cannot share here.
Okay, okay. We are just trying. So maybe meanwhile you can share verbally till we show you.
Yeah, actually we developed some peptides. Among them, I can show two peptides. One peptide is a very small peptide, which is an antibacterial peptide against the VRE, that is vancomycin resistant.
You can see your slides now.
Yeah. Next
so
one.
You go to the next slide now.
Next.
Next.
Next. So these are the previous one. The previous one, please.
Yes. So
previous one. Yeah, this one. We developed this peptide BF2. That is a very small peptide which is designed from cepacin V, that is antibacterial bacterial protein. So this shows a very good antimicrobial effect, good MIC values, and but with the combination with vancomycin, we can see it shows a very good synergistic effect, and with the ATCC and also the clinical isolates, and with the another antibiotic that is teicoplanin also shows a good synergistic effect, and with the clinical isolates and ATCC and other from the literature also we have seen that the peptides that other studies, these two peptides shows a very good synergistic effect. And another peptide, next slide please. Next slide. This is an antifungal peptide. Previous one, please. Previous one. Yeah, this is an antifungal peptide which is also showed a very good MIC value with the Candida tropicalis, and with this combination with the antibiotic amphotericin, it shows a good synergistic effect. and other with the clinical isolates also it shows a very good synergistic effect, but fluconazole has not shown with this peptide. The next slide. And from the literature also we have seen some peptides showed a very good synergistic effect with the combination with the antibiotic. So I can say that these combinations of the antibiotic and this antimicrobial peptide can overcome this AMR of this serious problem.
Okay. Thank you so much, Dr. Sharmistha. I will now address to the next question. Is Dr. Sunita Grover there? Yes. Yes.
Yes,
So,
yes, I'm
Dr.
here.
Sunita,
Yeah.
I have a question for you. What is the role of probiotics? In fact, we have already heard Dr. Panigrai's lecture. But what is your view? Can we use the probiotics? What is your experience in the prevention of MDR infections? And also, is there any role in today's era of COVID-19? Everyone asked that question. Can we use probiotics in some way? So, can you please share your experience?
Yeah. Fine. Thank you. I would like to share my slides. Can you see my slides?
Yes, yes, we can see. Yes, we can see.
So, okay, thank you, Dr. Rama Chaudhary. At the outset, I would like to thank Dr. Rama Chaudhary and her team for including me as a panelist. And in the 2 days deliberations, we have seen that there were excellent presentations by all these speakers on MTR and alternate therapies. So today, what I think is like 90 years back, when penicillin was invented, we could manage the infectious diseases with the penicillin. But now again, we are on the crossroads. We are in the era where antibiotic resistance is a great problem. So we do not know what to do. So we need to look for the alternate therapies. And Fajis, you have already heard Dr. Panigri talked about probiotics and gut microbiota. I will like to throw some light on the use of probiotics, the work done in my lab also. See, these probiotics, they are a novel and therapeutic option these days for controlling infectious diseases. They act as adjuvant therapy. Probiotics modulate the gut microbiota. In all the cases, whenever there is infectious diseases or antibiotic treatments, commensal bacteria are lost. So what probiotics do? They have to restore the gut microbiota. See, look at the gut microbiota of the normal people. See, it is highly diverse. There are commensal, there are symbionts, there are pathogens. When there are infectious diseases, what happens is the diversity gets low, see, and the gut is predominated with the pathogenic microorganisms, E. coli, Klebsiella, Enterococcus, and all. These belong to a category of microorganisms called probacteria. Our gut has got firmicutes, bacteroids, actinobacteria, probacteria, and variomicrobia. So what happens in this infectious diseases? Our gut becomes leaky. When gut becomes leaky, pathogens travel to the extra-intestinal organs and cause dysbiotic. We call it as a dysbiotic gut. So this dysbiotic gut needs to be balanced by use of certain other microorganisms like probiotics, fecal microbiota, etc. We had initiated work on probiotics a decade ago. First, the most important property of lactobacillus probiotic is to adhere to the human cell, our enterocytes, our cell lines. So in vitro, we tested and these probiotics adhere on the CACO2 cell lines and HT20 cell lines, and it was published also. We also studied the role of probiotics in salmonella-induced salmonella-induced infectious model, because when I was talking about the leaky gut, the gut permeability is lost, so permeability needs to be strengthened. So we wanted to look whether in the salmonella infection, what is the effect on gut barrier genes? So claudin, occludin, zonocludin, how they are being strengthened, how they are expressed, and how much gene is expressed. So based upon this, and we took certain probiotics and studied against MDR, MRSA, SORIAS, and ESBL, E. coli. First of all, I would like to tell what is the mechanism that these probiotics may act against MDR pathogens. One is colonization resistance. they confer colonization resistance. Probiotics, what they do is they compete for the nutrient and they compete for the receptor sites. So they lead to competitive exclusion of the pathogens. They form co-aggregation with the pathogens, and also they interfere in the signaling mechanisms. Then they have direct interaction on the bacteria, our pathogens, like they produce the antimicrobial peptides, bacteriocytes. Just now, peptides were given by previous panelists, and this reduced pH is due to the lactic acid, etc. And they have antibiofilm activity, antimicrobial activity. Second is the immune modulation. Everybody is talking about immunity these days in the era of COVID. So immune modulation is one of the very good property of probiotics. Third is the enhancing tight junction, gut barrier function, which I talked in my last slide also, that there is leaky gut. So we need to strengthen these tight junction proteins, which I already told, claudin, occludin, zonocludin, etc. And mucin production. These are the main mechanisms which I think probiotics can effectively work against MDR pathogens. So we took these strains from clinical isolates from Dr. Rama Chaudhary's lab. And first of all, we confirmed the presence of MRSA genes in the MRSA isolates, MECK gene, MECB gene, which are chromosomally encoded as well as plasmid encoded. And antibiotic resistance profile also we confirmed. Similarly, ESBL isolates were also tested for chromosomally encoded genes or plasmid encoded genes, and were also confirmed that whether they are ESBL positive or negative by using the antibiotic plus inhibitor assay also. Then we took live probiotics and surface proteins also. See, now the probiotics, when we are talking about leaky gut and all in critically ill patients or immunocompromised patients, these live bacteria may lead to translocation and cause bacteremia. So if we take postbiotics, which include certain metabolites, or which are the cell wall components, we call them as MAMPs, molecular patterns, microbe-associated molecular patterns. They recognize the PPRs in our intestine and lead to the signaling mechanisms. So we here worked on surface proteins. So we tested these our isolates, probiotic isolates with MRSA strains. They form co-aggregate. That is, co-aggregation was very effective with some of these strains. And antimicrobial activity, you can see here, they also act as antibiofilm formation. We studied adhesion on mucus also. In mucus, competitive exclusion was there with the MRSA strains. And also we did anti-colonization in taking HT29 as cell line, wherein we took two of our strains, LF1 fermentum and A5 and then two of the reference strains, and studied the protective, competitive, and displacement assays. It worked very well against the SRS. And we also did transwell plate assay using HT29 cell and with using FITC dextran. And we found that gut barrier function is also getting restored. Similar thing was done with using ESBL. So there also first we screened all the probiotic strains, all the E. coli strains, selected 3-4 E. coli strains and 4 probiotic strains, and again did all the assays which I elaborated in my previous slide. And here this shows that in competitive co-stimulation and displacement assays, the probiotics were very effective against these ESBL strain. Here the strain was different. One was fermenter, of course, the other was LBS4. And similarly here also we took we did transwell assay. So we found that these probiotics and surface proteins act very effectively by inhibiting the adhesion site, by competitively excluding the pathogens and improving the intestinal barrier integrity. So what we can do is, because we cannot eliminate antibiotics at all, because that are very useful for the critically ill patients. We need to give the antibiotic, but we need to use probiotics as the adjuvant therapy. It will lead to faster healing. We can reduce the dose of the drugs. And if we use the probiotics, it will reduce the side effects of the drugs also. Increase the eradication rates of microbial infections. Reduce the risk of antibiotics-induced superinfection in the gut. and generally they are safe and well tolerated. However, the dark side is that if there is translocation and we are using in immunocompromised or critically ill patients, these probiotics may translocate to extraintestinal site and lead to systemic infection, bacteremia, endocarditis, pneumonia, etc. And the other thing is, because our gut is a reservoir of antibiotic resistance genes, so they may transfer these genes if we are using long-term probiotic therapy from pathogens to probiotics also. These genes will be transferred. And when we are using the probiotics, we need to look at the resistance profile in the probiotics. And most important is whether it is intrinsic resistant or extrinsic resistance. If it extrinsic acquired resistance, we need to look at whether it is chromosomally encoded or plasmid encoded. That is very, very important. And the other strategy is the fecal microbial transplantation, because now we know that it is our gut which is responsible for our health status. So if in CDI it has been used extensively, and there is 75 to 94% recovery rates, because antibiotics kill all the commensal bacteria also. They are not selective in killing only the pathogens. So commensals are also depleted out of our gut. So here in fecal transplantation, the stool of a donor, is a healthy donor, is taken and transferred to the diseased person, where the gut is first made sterile, and then delivery is done through enema or the nozzle or gastrotubing, etc. But only thing is, greatest disadvantage is this, that we need to be again very, very careful that the donor does not transfer any kind of pathogen or any kind of disease causing species. So we need to be very, very careful.
So fully agreed. Thank you so much, Dr. Sunita. And I think this cautious.
COVID.
Yeah, please.
Second question, COVID.
Last
COVID. Just 2 minutes more I will take. So I would say that bacterial resistance is inevitable. It's an evolutionary process. Because probiotics are generic effects. So we need to design designer probiotics. I read a paper wherein they have designed a probiotic which contains P-cure plasmid. That plasmid, when given, that probiotic when given, that plasmid stops the replication of the antibiotic resistance plasmid and targets the specific genera of the pathogens. So those kind of things need to be carried out. So coming to COVID-19, mechanism remains the same, like probiotic, how they act, which I have already explained. COVID-19 acts via the respiratory route, but its contribution of gut towards pathogenesis is also cannot be ruled out, because COVID RNA has been detected in stools of the patients, and we know that receptors ACE2 receptors are very common in lungs, so that's why they attack lungs. But they are also expressed in the gut, so that is why COVID also is in the gut. So this probiotics, we have already, it has been established by several of the workers here, several of the reports, that it is very effective against respiratory tract infections, lower, upper, ventilator associated pneumonia, etc. So like we always say, there is a gut-brain axis. So there is a role of gut-lung axis also. So if we take probiotics, see, this is the cos virus, these are the probiotics. If we take them, and then short-chain fatty acids are produced, leukocytes, there is increase in phagocytic activity, leukocytes, they are all translocated to the lungs, and their lung also microbiota gets balanced, as well as the intestine, and it's a bidirectional action. But before taking in the probiotic, none of the studies in India have been done on the gut microbiota. There are 2 Chinese papers which I could get. This is one paper wherein they have taken the stools of the patients who were hospitalized for during the whole stay. Every week they were taking 2 to 3 times these stools. They found that commensal symbionts, they were decreased, like Faecalibacterium rosebury, because these are anti-inflammatory probiotic bacteria, and their presence was depleted. And the opportunistic pathogens, there were high number of opportunistic pathogens. This is one study. The another study is this, that if you see the healthy gut, these are the bacteria, and COVID gut, these are the bacteria. So Chinese people have done this, and they have made a probiotic preparation also to give it to the COVID people. Bod et al. Recommended some of the probiotic strains which are used in respiratory infections, and they have recommended these can be used. But in my opinion, only rationale for use of probiotics in COVID-19 is indirect evidence from the reports which we have already, the reports have been generated. So we cannot recommend random blindly that conventional probiotics can be used unless until we see what is the effect of COVID in the gut microbiota, how our gut microbiota changes, how the pathogens are overwhelming and commensals are decreasing, then we can only look for certain probiotic strains. But these might be useful for secondary infections. So future of probiotics is at present not very clear, because we need to have a specific strains of probiotics, and we must do certain clinical trials so that we generate the data on the efficacy, that how probiotics are good for COVID. With this, I stop here. Thank you so much.
Yeah, really, it was wonderful, because people have always this question about COVID. So Dr. Vinod is back here. Dr. Vinod, are you there? Okay. So would you like to first address this question and then go to your presentation? How you want to do it?
Whatever
I
you
have a question for you first. That
Yes,
you have done some good work on bacteriophages to clear the sewage system. So I wanted to know from you whether we can use these phages, and how effective they will be if we can use in the cleanness of sewage system from the MDR bacteria. So please share your experience in this.
Thank you, ma'am. After the panel discussion, it will be a great honor, if I could make a presentation on chimeric phages. So
yes,
ma'am. I'll share my slides. Can you see my slide, ma'am? Can you see the slides?
I can see this. Yes, I think it is there. Yes.
Yes, ma'am. Of 20 years of working on phages and looking into the regulatory issues, I felt phages are more suited to sewage. I collected water from 3 busiest hospitals and characterized the bacteria. So I could be able to isolate the Enterobacteriaceae members like E. coli, Enterobacter, Pseudomonas, Acinetobacter, Citrobacter, Salmonella entritidis, Proteus mirabilis, Coagulase negative Staphylococcus, Enterococcus faecium, and Enterococcus faecalis from all the hospitals. I've labeled them as hospital A, B, and C. And majority of these gram-negative bacteria, they were pan-resistant to all the drugs. They were resistant for more than 7 classes of antibiotics. This was before the treatment. This is the sewage treatment plant. What they do is initially they do the pre-treatment and the primary treatment, and it goes to the secondary treatment and the tertiary treatment. So in the tertiary treatment, they use the chlorine to disinfect the sewage water and also the hospital effluent. And usually they use around 4 ppm of the chlorine to disinfect the hospital effluent. So the bacterial count initial and after challenging with the chlorine at the different time interval. So initial bacterial count I've given, and I have cultured after exposing to the chlorine at the interval of 2 hours and 8 hours. So in case of hospital A, ppm of chlorine was sufficient to kill the few bacteria like Klebsiella pneumoniae, Enterobacteriaceae after 8 hours in case of hospital A, whereas in case of hospital B and hospital C, even though the bacterial count decreased after 2 hours after exposure to the chlorine, but the bacteria, we could able to isolate the higher number after 8 hours of exposure to the chlorine, indicating that majority of the bacteria are resistant to the chlorine. Fluor and drug resistance bacteria are almost everywhere, even the PET bottle or the potable water what we drink also contains the, the drug resistance bacteria. Even after effluent treatment also, the water which is released into the community or into the river, it contains drug resistance bacteria after challenging with the chlorine. So this we could able to isolate the bacteriophages specific to all the bacteria which were isolated from the wastewater or the sewage water or the hospital effluent. We could able to characterize them, do the electron microscopic study, and at the tertiary level, we could able to add 500 ml of phages into the tertiary unit, and after 2 hours, 8 hours, and 16 to 24 hours, when we cultured it, the bacterial concentration start decreasing. We didn't isolate the bacteria after 16 hours in all the hospital, indicating that bacteriophage are really effective against the bacteria which are isolated from the sewage water and even the hospital effluent. Just comparison sake in this slide, the chlorine get easily oxidized over the period of time. When you keep the chlorine for 8 hours time, it get easily oxidized. What is important here is a definite concentration of chlorine should come in contact with the bacteria. So over the duration, 8 hours to 16 hours to 24 hours, the concentration of the chlorine decreases due to the oxidization. Whereas in case of phages, there's no need to add extra chlorine to the tertiary treatment unit because the phage number increases. That's the, the curve which is showing. So the phages were very effective because I heard around 6 speakers yesterday and the 7 speakers today speaking on the alternatives and the strategy. The phages are most suitable, agent because of the regulatory issues. Thank you, ma'am.
Promising approach, and this may be very, very useful in today's era. We also have, we will have your presentation after the panel discussion. I just want to ask Dr. Gopalnath is there, and he has immense experience in the case of bacteriophages. The challenge, Dr. Gopalnath, I want to ask is that most of the people are having problems from the regulatory authorities, and although we have, many of us have isolated phages, but when it goes to whether we can use it in clinical settings, how to go about it and what is your experience and whether we can really use it or we have to take permission from certain DCGI or the FDA or whatever. So can you please enlighten us on this aspect?
Okay, ma'am. Thank you very much. So in this regard, I tried to meet the health authorities. You are aware about that one also. So they suggested that maybe we are going for more animal preclinical studies to prove that this is really safe to use. So for that reason, for last 1 year, I just went for 2 things. One is that orally it is not causing any problem. The data also says the same thing. In rat model, we have given even up to the 10 power 20 PFU per rat orally every day for 6 weeks. Nothing happened. And then the other, we just injected the bacteriophages subcutaneously for 6 weeks, every week once in rabbit model. And we found that the neutralizing antibodies are rising at the end of the 4th week, usually after 3rd week. So if acute infection is there, the neutralizing antibody is not a problem. And oral use, I feel, is quite safe. So if we are able to convince the regulatory bodies, probably they must permit us to use, and then on compensate ground, we must also permit for using this, because already we have done the bacteriophage therapeutic effect on the septicemia model, which we encounter in the ICUs, especially with the bacteria like Acinetobacter, Pseudomonas aeruginosa, and Staph aureus, and Klebsiella pneumoniae. Recently, we have concluded that. So it is safe basically, but they say that there may be some modification in the host, this microbiota that may be causing some problem. But if you are using the bacteriophages which are in a cocktail form, So they are basically eradicating all the host bacteria. So transfer of the genetic material is not that great issue. The other thing is that we have convinced that the bacteriophages are microbiota of our body. That includes the gut microbiota. So this will expect that it will induce the antibody formation. So that is not happening. It means somewhere our immune system is recognizing it as a commensal, a part of the commensal microbiome. So these are the issues. Probably we have to work more, and we have tried to convince them that they must permit us for using these things. Thank you.
So I think it's a very pertinent issue because Dr. Taruna showed she has a good Faj library, and with the 101 posters, which many of you may not be knowing, which came to us, we had large number of posters on Fajes. So definitely many groups are working which we are not even aware. So I really feel that Dr. Gopalnath, especially as a secretary of the Indian Bacteriophage Society, should really now make an effort and make a consortia where we can pool these phages, and rather than getting it from abroad and treating our patients here, I think we have a very good collection here. We have Dr. Vinod's phages, Dr. Taruna, Urmi is working on phages, and the group in Calcutta, which I've discovered recently, is working on wonderful phage collection. And I think if we can pool all these phages, we have very good collection ourselves, and then we can share and use it for the maybe human use. And one thing which he has shown very clearly is if it is in the superficial wound infection, I think even if we can do that itself is a very big breakthrough, because so many people with diabetes or DVT or thrombosed veins are having these ulcers and they are suffering. So I think we should immediately meet the government authorities, ICMR, DCGI, government, whatever, and start this kind of approach for the treatment at the earliest. We have had a meeting earlier where Dr. Gopalnath was there, and I think the one of the aim of this webinar was to be able to create that body which is already there, but to be able to work together with them. So I think if we can do that, it will be wonderful. Then it will be really useful for the people. And why only phages? We can even think of having combination of phages and probiotic, as some of the slides were showing, or maybe even antimicrobial peptide, or maybe some portion of the probiotics. I think that will be very useful approach. Is Dr. Martha there? I wanted to ask her some question. Martha, are you available? I saw
Yes.
you briefly. Yes. So I want to ask you that what kind of permissions you need in your country, because in India we are facing this problem, and what all kind of regulatory formalities you have to do before you start using it in clinical application, especially for C. diff. We have there's a lot of scope, and it's already a super bug. So can you share your experience regarding this?
Yes, of course. In the UK, we have a very similar regulatory problems, or not problems, but it's very similar pathways to overcome. And I would say that, interestingly enough, in the UK, I find that the people that regulate veterinary products and animal products, I find are more willing to engage in a dialogue. So that's why over recent years, I've also been developing phage products to treat so pigs and chickens for salmon, and those regulators they're coming to the conferences and they're really telling us what data we need, and the medical regulators are starting to do that as well, but it's quite it's quite a long process. So one thing that we're trying to do at the moment is map the areas that we focus on to direct clinical need. So I think we'll have so there's quite a bit of scope for using things where we can show there's no other option. So if we can start there, so if we can use phages to treat the sort of the urgent clinical need where there are no other options, the regulators are more sympathetic to that. So we collect, that's the plan, is to try to map that need, because to where the pathway will be easier, because we have a situation probably similar to you in India, where we have people basically the doctors are communicating with me directly saying, I can't treat my patients are dying. And then I'm in a position where I can't just give them the phages because I don't want to, we still need to collect more data and we don't want to break any regulations. So it's a question, I think, of narrowing, working out what data on safety, particularly, that we need to narrow that gap. So I'm working both with, interestingly enough, I'm working with public health doctors as well as to try to make that bridge tighter.
So do you need any permission if I'm, say, for example, like if we are getting some strains from abroad, we have to follow certain regulations and rules. So supposing we are getting phages from abroad, from UK or Russia or whatever, do we need permissions, or do you have to take permission when you are sending these phages to other countries for the therapeutic uses or for the clinical or research use maybe? So what kind of permission you have to take, or how we have to go about it? Like supposing we have to send these phages to some other collaborator or some other physician.
Yeah, I think sending phages is in a way easier than sending bacteria, because you're not sending anything hazardous. So you can show that I can make very nice pure preparations of my phages, which then don't contain any toxins or anything hazardous. So that's okay. Most of the phages in my collection, I've isolated them. They're my phages. They're not owned by anybody else. So there shouldn't be restrictions on sending them, which I'm very open to collaborating and working exactly like that. I mean, the main thing that we'll have to do is you always need to check the specificity of strains in different countries. So, for example, I have very nice projects with Thailand. So it's for for salmonella, and then some of the phages we have in England are very effective on the Thai strains. But then for others, we needed to find phages in Thailand. So my guess is that for India, it will be a mixture. Probably some of my phages will be useful for your strains, but we may need to help you show you how to find other ones to fill in those gaps. So I think it'll be a mixture. But no, actually sending phages is not so difficult. Oh, sorry, I think it's on mute. Is the microphone on mute?
Yeah. So what I'm saying, that this is great to hear, because most of the people have this fear whether we can get the phages and when we can collaborate. So I really feel that if it is not a big problem, I think there is a need where we can share the phages, and once we know they are specific, they're fully characterized. Because what I understand from your talk is that you have actually fully characterized these phages as far as C. difficile phages are concerned, and we are sure that it is mostly lytic phage and there is no kind of integrase you were mentioning, and maybe they are not lysogenizing phages. So once they are characterized, once they are specific, then I think it's the right time to use it and share with others. And also I want to know, do you have any phage bank? Like Dr. Taruna shared that in India they are maintaining phages. In Hisar we have a small phage bank. In fact, I have seen personally, it's a beautiful phage collection and beautiful way of storing bacterial strains. Very, very rare strains from animal collection usually, but which you may not find anywhere in India. So my question to you is, do you have any bacteriophage bank in UK or in your this thing, in your guidance, whatever?
Yeah, there are no sort of official phage banks. They're probably mine is one of the largest ones, because I've been working now for nearly 2 decades with different phages. So we have nice collections, which we just store them in multiple -80 freezers. We have some of them we bank in other places as well. So some of them I give to other collections. For example, Public Health England have some of my phages in their collection as well. So yeah, so I do have a sort of fairly well curated bank with multiple backups in different places.
So that will be really wonderful. And as you are already aware, because you have given a talk in the Bacteriophage Society earlier, where I listened to you people. So what I said was that Dr. Gopalnath will be in touch with you, and we will see how best we can use. We can collaborate, and the idea is that how best we can use these phages, especially in this era where we are wasting so much of time looking for new phages. Rather than that, those who have, they can share and we can mix and we can see what best combination can be used. whether it is India or UK or Russia or whatever. So with that, I would also invite, we have, do we have Dr. Ramesh there? I don't think he's there.
Yeah,
Dr. Panika, Ramesh, you are there? Okay.
Yeah, I'm here.
So, yeah, Ramesh, Dr. Ramesh, I wanted to ask from you. You have wonderful papers. I've gone through your papers. It's a beautiful collection of the work. But what I want to know from you is, have you used any phage in a clinical application, or this is still in the basic mode? I mean, have you applied your phages in any clinical application so far?
Yes, we didn't apply directly to the clinical application. So we drew and also, since because of the finance constraint and other things, and ethical approval because of that, we didn't do that. But they are ready to, and they are discussing with the team now. I hope we will be testing our pages with the team.
So have you deposited your phages anywhere, or they are still with you? Have you deposited it officially somewhere, or
yes, we did the whole.
Okay.
NCBI.
So you have submitted on to the gene bank the sequence. Okay. Right. So is there any? Do you have any information?
Maintaining a page bank, more than 60.
Okay, wonderful. So I just want to know: are you? Does NCCS Pune maintains any phage collection? I mean, I'm not very clear. They do maintain MDR strains,
Yeah.
but is there any information you have whether they are maintaining phage collection there?
As far as they are maintaining the MDR pathogens, I didn't know that they are maintaining the bacterial pages. Yeah.
Okay. So thank you, Dr. Ramesh. It was wonderful having you. And
thank you so much.
yes. So is Dr. Pinaki there by any chance? Dr. Pinaki Panigai, have you logged in? Because it was early morning in Washington. It is, I think, 6 o'clock when I asked him to be awake and participate. So if he has not logged in, I will request meanwhile, Dr. Vinod, if you can we can have your presentation, and then we have to summarize and announce the awards. And I'm sure many participants are waiting to hear their result. So Dr. Vinod, can we have your presentation now? Thank you so much for the wonderful discussion.
Yes,
in the lack of time, I have not taken the students' question. I will see if it is possible, and after your presentation, we may take that. So please show your presentation.
Please. Yes, slide. Can I share my slides, please? I'm trying to share my slides. My slides are not shared, I think.
It is sharing now.
Now, okay. Okay. Give me 1 minute, please. Let me share the slide. Internet issues. Yeah. Thank you. Thank you very much. Because yesterday I just speaking on much to share. The topic here which I applied for patent, and I'd like to share a very few glimpses of the work what we have done. It's a very projecting slides which shows the highest number of articles published on antibiotic resistant EOR. So it is mainly from 2015, the highest number of publications coming from UK, Netherlands, Belgium, Germany, Liberia. The highest publications are from India, and suggesting the serious implication and the effect of antimicrobial resistance. Madam, can you hear me?
I know the slides are not visible, so maybe you can share just like that. We are not able to see your slides, but if you can share, your major findings. Because they're not visible. Yes, yeah, please
slides
share
not,
major findings.
Yes, you can see the slides, ma'am? No?
No, they are not
Okay,
visible.
the slides are not visible. I don't know. Okay, okay. Not an issue. Maybe it's connected. Okay, then without the slides, can I present, ma'am? Is it okay?
I can't see.
It's okay. Can you hear me, ma'am? Can you hear me? Can you hear me?
We can hear you. We can hear you. You can speak and tell your major findings.
Okay, ma'am. Okay, fine. The billion-dollar question here is, what came first? Is it a drug resistance or antibiotic?
Dr. Vinod, I think you can switch off your video, then maybe your voice will be audible to us. There is a lot of network issue at your end.
It's quite audible.
The main hurdle in the fight therapy is the need for a fundamental knowledge of interaction. Is it audible, ma'am? Is it audible? Ma'am, is it audible?
Yes. Yeah, you are audible.
Yeah. So shall I continue?
Yeah, please continue.
Thank you, ma'am. Thank you. The most important fundamental hurdles in the fight therapy is there is no fundamental knowledge of phage-bacterium interaction in the animal model before we pick up the clinical application, or it can be envisaged for the fight therapy. The main issue here is the species specificity missing of the bacteriophages. The difficulty to include high specificity rendered bacteria targeting only few strains for the given species is one of the most hurdle, and also the sterility and purity of the batch preparation. And genetic modification of the host rendering it virulence and the pathogenicity of the host. And as Dr. Mata was stressing, regulatory challenges. In the past, we have developed various animal models, like septicemic model, diabetic model, and in the rats and rabbit, burn-on model, meningitis model, UTI model, and also the bacteriophage in wastewater treatment. And the most interesting aspect is the bacteriophage in the treatment among the biofilm-producing bacteria in the ventilator-associated pneumonia. Also the chimeric phage, which is the answer for the genetic modification of the host. So a lot of bacteria, gram-positive bacteria and the gram-negative has been implicated as the etiology of ventilator-associated pneumonia. So what we've done is, since we cannot use natural or the wild bacteriophage as a therapeutic agent, we thought of utilizing a bacteriophage T7 lambda phage, which is a lytic phage of the Escherichia coli, which is an important model system in the molecular biology. The main reason for selecting the T7, because it has an advantage of having a relatively small, completely sequenced genome, and none of the gene code for toxins here. And it also can replicate in the slow-growing other starved bacteria. Utilizing this, what we did is we exchange the C-terminal receptor binding domain of the different phages, and they will fuse with the N-terminal phage binding region of bacteriophage 7, so that you have a chimeric bacteriophage, the head of T7 and the tails of the bacteria of your choice. So we selected 3 important bacteria which I wanted to show. The Staphylococcus aureus, which is methicillin resistant, and extensively drug resistant Pseudomonas, and extensively resistant Acinetobacter baumannii. The chimeric bacteriophage along with the hydrogel into the endotracheal tube, and we sprayed the bacteria onto it, and we cultured the bacteria at a different interval. Within 6 hours, the bacterial load significantly reduced. There were 5 log reduction in bacterial load within 8 hours, and around 2-fold reduction 8 hours, and no bacteria were isolated after 24 hours by using the chimeric phage. The aspect here was we compared the effectiveness of the chimeric phage with the wild phage and also with the silver nanoparticle. We use silver nanoparticle of this diameter of 7 to 15 micrometer size and strength of 20 micrometer per ml, and it was sprayed around 1 centimeter square of 20 microliter. So we compared with the silver nanoparticle, one group. Second group, we compared with bacteriophage, the wild bacteriophage, and then we clubbed the silver nanoparticle with the bacteriophage, and we compared whole result with the chimeric phages. Silver nanoparticle with a of 20 microgram had a bacteriostatic property, because the moment to enhance the strength of the silver nanoparticle, it becomes cytotoxic. So you can't use above 20 microgram. At 20 microgram,
Dr. Vinod, can you summarize the key points at the end without discussing
yes,
the details?
Yeah. Yes, two more minutes, ma'am. 2 more minutes. With the silver nanoparticle, it is more of bacteriostatic. But when the same silver nanoparticle is clubbed with a bacteriophage, we observed a very significant reduction within 16 hours in the endotracheal tube. But when the chimeric they used individually, the result we could able to see the drastic 7 log reduction in the bacterial count. Chimeric phages were clubbed with silver nanoparticle, the endotracheal tube. We did an study, and we found no biofilm formation was in the endotracheal tube with silver nanoparticle coated with the chimeric phage. The advantage of using chimeric phage is adaptable to the different bacterial strain, and also the phage matrix has to be safe, and in small protein domain on protein not change the safety profile, may significantly change the range of bacteria may be successfully infected. The silver nanoparticle with the very effective against the attachment of both brain-positive and gram-negative bacteria. It is prudent to proceed with the 5-10K as a convincing data on the safety and the efficacy of the public domain. Dialogue with the regulatory bodies, highlighting the advantage of the phages and the issue related to the the gene transfer among the host and phages, can definitely take bacteriophage as an alternative therapy where the impact of antibiotics are fading. Thank you, Dr. Rama, madam. It's unfortunate that the internet connection is not proper, otherwise I would have shown those. Thank you.
Yeah, so we are also sorry for that, but then please, in the YouTube, we will try to upload your slides if you are able to send it. So I'm sure at the end of the panel discussion and all the talks, many of you must have been waiting for the ASM award. So I must tell you, we received a very good response for the participation with the e-posters. We had 100 e-posters in 3 different categories. And these categories were antimicrobial resistance, infection control and prevention, and alternative therapy, and including bacteriophages and nanoparticles. So we had 14 posters from East, 48 from North India, 29 from South India, 6 from West India. We have 72 posters in antimicrobial resistance, 13 in infection control and prevention, and 15 alternative therapy to combat AMR. All the posters were so interesting and they showed very good quality of work by the participants. 4 esteemed national reviewers and judges and 1 international judge evaluated these posters. It was a real difficult decision to select just 2 best posters out of it, and which will be given award from the American Society for Microbiology worth $150. They usually give the membership and the ASM books as an award. And so what we decided, we really wanted to appreciate much more than this, and we are also giving certificate of appreciation and award in addition to ASM award. So I will now first announce the ASM award. The first award goes to poster number 46, and it is 189 registration. Can I have the snapshot of the awarded poster? This is Dr. Swati Sharma. She's from Department of Microbiology, Banaras Hindu University. Her topic was mutation at novel sites in PMRA/B and IPXA/D genes, and no compensation in fitness in colistin resistant Acinetobacter baumannii from a tertiary care hospital. Congratulations to Dr. Swati. I don't know whether you are already there. And it is a wonderful poster. Congratulations for the ASM award. I'm sure Dr. Pal is there, if not the student, and we can congratulate you. This is your university which is getting an award. The second award goes to poster number 76. The student registration number is 565. And so that shows that more than 500 participants have registered. So the girl name is Dr. Rituparna Tiwari. She is from ICR, National Institute of Veterinary Epidemiology and Disease Informatics, Bengaluru. The topic of the poster was frequency and genetic diversity of extended spectrum beta-lactamase, MC, lactamase and metallo-beta-lactamase producing Escherichia coli from human and animal populace, a One Health molecular surveillance approach. So this was another wonderful poster which is talking about One Health, and congratulations Dr. Rituparna, and congratulations to the whole team. And I think it was a huge team. They have collaborated with some international collaborators also, and I think the teamwork always wins. So congratulations to this team. Now, in addition to this ASM Best Posters Award, we are also honoring the certificate of appreciation to the many other participants who have done a very good job. And the first one in antimicrobial resistance is certificate of appreciation goes to Dr. Pushpa Kumari Nirvan. She is from Artemis Education and Research Foundation, New Delhi. And her abstract number, poster number is 83. Her registration is 199. The topic again is similar, mutation of 4PQ PMR AB2 component system in colistin resistant Klebsiella pneumoniae, report from a North Indian tertiary hospital. And this is another wonderful paper. It was very difficult to decide whom to give the award because most of the colistin resistance posters are of great importance. The next poster award of appreciation certificate goes to Dr. Ronak Beer. He is from Department of Microbiology, AIIMS, New Delhi. And his poster number was 35. And it is colistin susceptibility testing and MCR1 detection for carbapenem resistant enterobacteriales. Comparison of 4 phenotypic tests with broth microdilution. So this is another very important aspect because not every lab can do the molecular test, and we should have the other testing methods available which can be done in other labs, and these phenotypic methods still remain important, especially in our country. And this is another poster award which goes to as a certificate of appreciation. Then in the next category, congratulations to both of them. Then the next category is on infection control and prevention. Here the first award goes to Dr. Raksha K, St. Martha Hospital, Bengaluru. and the poster number was 28. The registration number is 296, and the topic was very catchy. It was Double Trouble, Antimicrobial Resistance and COVID-19, St. Martha Hospital, Bangalore. Congratulations for the wonderful poster. And all these posters are available on the website. If you click the e-poster link, you will be able to see all 101 posters there, and they will be available because the website is continuing for at least a year, and they will be available all the time. So the next one goes to again to Banaras Hindu University, Dr. Tuhina Banerjee, for setting up of infection prevention and control practices in an upcoming COVID hospital, an experience from North India. And this is 88 poster number, and it is 237 registration number. Congratulations to both Raksha and Tuhina for the wonderful award. The next award goes for the bacteriophages work, and this is another very good work. The poster number is 38. The candidate name is Dr. S.K. Tausif Ahmad, Acharya Prafulla Chandra College, Kolkata. And they have characterized 3 novel lytic phages infecting multi-drug resistant Shigella species and E. coli. And I must say, wonderful work. They have characterized phages so beautifully. You will see the poster. I really want all of you, whether you have been awarded or not awarded, please go and see these posters and learn how to make the posters also. And this work was wonderful work, and they have elaborate, extensive studies they have done on these phages, going to the genome level and doing all characterization properly. The next award in the alternative therapy categories go to Dr. Basava Prabhu HN. He is from ICR, National Dairy Research Institute, Karnal. The poster numbers were 44 and the registration number is 511. The title is biosurfactant of lactic acid bacteria, the potential weapon to tackle the persistence of clinical isolate of methicillin resistant Staphylococcus aureus. So congratulations to Dr. Ahmed and Dr. Prabhu and Dr. Sunita Grover. I think it is her team, Dr. Rashmi, for wonderful work. Congratulations once again. Then we have 2 more awards, and this is a special category we have to create. The reason was that they are not typically falling into any particular category, but these 2 awards have been given for the novelty of the work and for the innovation. And the innovation was poster number 53 by Dr. Divya Anthwal, Translational Health Science and Technology Institute, Faridabad. She has developed, it's a whole teamwork. They have development of biosafe filter technology for sputum transport to enable direct detection of drug-resistant TB in sputum. So this is a very wonderful work, and I'm sure they must have already got the patent and must be in the commercialization phase. So congratulations to the big team, and I think there is also a TB hospital faculty I could see in this work, and I congratulate the whole team for the wonderful technology they have developed, and I'm sure this will be going very soon into the commercialization. Last but not the least is the award for Dr. Colony. This is one field which is hardly done in India, and that is anaerobic infections. And this is from Father Muller Medical College, Mangalore. And the poster number was 13. The registration number is 333. Lucky number. So she has been given recent award for the recent trend in antibiogram of Bacteroides fragilis group and resistant genes from clinical specimen. So I know that it is very difficult to work with anaerobes and then to do something with the antibiotic susceptibility, looking for their genes, and they have tried to look the whole spectrum of the genes in these bacteria. Congratulations to Dr. Colney and to her guide, Dr. Beena from Mangalore, once again. And the good news is to come still. All this certificate of appreciation will also be awarded a cash prize. And this cash prize is of 5,000 for the first award, the name which was announced first, and 3,000 rupees for the second award. This certificate of appreciation and the cash award will be sent to you in due course. And I think we must appreciate these young people's hard work. And I must tell you, all the 100 awards, if I could really give, I will give award to all 100 posters, because we announced the poster announcement just a week before the webinar, and within a week we have got these posters. So I think everyone must have worked very hard. There are many which we were really good, and it was not possible to get so many awards, but definitely I must say that the quality of work is very good, and I'm really proud that Indians are doing wonderful job. And please go again to these e-posters. I will request all of you to click the posters and see what best work is available, and we can compile it and put it in a kind of, we are releasing a book after this conference. We will be including all the posters, all the presentations, and messages which we have received. In fact, I don't know, many of you must not have seen, we have received a message from the health minister, Dr. Harshvardhan ji. We received the message from Dr. Vinod Paul, and also Dr. Guleria, and many others, but these three are already included in the website. So congratulations to everyone. And before I end, it will be wrong if I don't fulfill my responsibility of thanking my team. We are all the time seeing me, but actually there are people who has worked with me to make it a success. I want to invite them here quickly. Dr. Nishan, Dr. Hitinder, Dr. Sarita. So I will quickly tell, in addition to Dr. Bimal, who is co-chairperson, and also the treasurer, we didn't have to do much of treasurer work this time. But I will say that Dr. Hitinder, please come here. He has been coordinating with the ALCOD, and all the the dirty work he had to do, coordinating with these people, trying to get the presentations, trying to make the designs and things like that. So thank you so much, Dr. Hitendra. I want to invite Dr. Gagandeep. Dr. Gagandeep is the one who has been sending you all the emails from AIIMS ASM, and it was not Dr. Rama, it was Dr. Gagandeep. So thank you so much. Then we have Dr. Sarita. Please come here. Dr. Sarita has been all the time telling me for the e-poster. She designed all the the framework for the e-poster. Within a week, she was really working hard. And every night, we were initially saying only 12 posters I have. So I said, okay, no problem. And then 20, and then 30, and 40, and 50. And the last day, when we actually concluded, it was 101 posters. So it was wonderful response which has come from the people. And Sarita has worked very hard to coordinate with the judges. I initially thought I will announce their names, but they have requested for the confidentiality. But I must say that one of the judge at 3 p.m. at night yesterday told me that he has completed the evaluation. So they had to really work very hard for it. And other persons were like giving us in batches, that because every time they have to really take note of it. And thank you, Dr. Sarita, for wonderful work. And without you people, I could not have done. Dr. Nishant, the most silent person in our team, but he is always, I have to fall back on him. Dr. Nishant, this thing, I will put my. So he is the one who is like all the troubleshooter. So when others are not able to give me a solution, I fall back on Nishant. And for making wonderful, contribution to the whole program. And I'm really thankful to all these four people who have really contributed immensely. Perhaps you people will not be knowing that it was a conference to be held in April, on 3rd April. And then we postponed it to 3rd July. We did not know pandemic will continue. And then we have to, from 3rd July, we have to postpone it now in October. So they have been working since almost 6-7 months now for this particular webinar. We also have Dr. Priyam with us and Dr. Swati, and Nisha is here. Nisha, please come here. So Dr. Priyam has been coordinating today, and also we have Dr. Swati. She was our MC and doing every innovation and everything. Thank you so much. And also we have Nisha with us. She is working on bacteriophages. I could not present my work because she has actually prepared her present. Everything was done. We are really sorry. Dr. Rashmi's presentation was also ready with us because we were not sure whether Dr. Pinaki will be able to log in at the last minute in the morning hours. So we have kept the backup. But I must say that Dr. Rashmi had beautiful presentation on the zystome, and she has also prepared where we have made a kind of combination of bacteriophages as a surface disinfectant, and that is something which we are trying to use in our hospital for the surface disinfection. Thank you, Dr. Nisha, for your hard work. Where is she? Please come here quickly. So, yes. So she is my, main person working on phages, and she has actually coordinated and gone to Dr. Gopalnath's lab and learned a lot from there. Also borrowed some phages from there, and also Taruna held a workshop. She also attended that, and maybe to Dr. Ramesh. So she has been going to all the workshops and seminars. So last but not the least, I want to thank ALPCOD. We have one representative from ALPCOD here. The event management system person who has who was really bored from the morning till evening. He had to sit because he may not be understanding anything. But all the time I'm scolding him that voice is not clear, the slide is not clear. Thank you for your patience, and it was a wonderful work your team has done. I really thank you and congratulate you. We also have our own person. आप भी आ जाइए इधर. This facility is in Convergence block on the second floor. It is called SET facility, and I don't know other people are there, but so he has been coordinating today, and there are other SET staff. I really want to thank them. They are not here today. I think they leave by 5. They have been a great help to us, and even yesterday everything was worked out nicely. Sorry for the whatever glitches we had today and yesterday. This is the sometimes disadvantage of the using digital platform. But thank you so much. And last but not the least, we also have some attendants who have been running here and there. You can also come here. Come. Because we always feel that we need water, we need coffee. You may not be knowing I had 2 cups of coffee in between, and he is the person who has been getting it. Batao apna name. Prashant. So he is a lab attendant and recently joined, but I want to thank him also and many others who are not here today. Sumit was there yesterday. To all my team, to whole my department, and to the AIIMS, because Dr. Guleria gave wonderful, message yesterday, and NCDC director gave me a video, and even the health minister office gave us the message last minute when there was a very important meeting going on. So I'm really thankful, starting from the government, starting from WHO, CDC, receiving response from ICMR and from AIIMS, especially from our director, Dr. Guleria, our dean, Dr. Anita, and our research dean, Dr. Subrata Sinha. Thank you so much. And we can quickly, all of us who can open, have their videos on, we can have a quick picture. Those who have logged in can have their pictures, videos on, so that we can see all of you. Ranjana, if you are there, you can put your video on. I can see Taruna. Martha, if you are there, yes, please have your video. Yeah, Ranjana, thank you so much. Sorry, we had to cut short your this thing. And but it was a wonderful having your talk today.
Yeah, thanks for having me, ma'am.
Yes. Taruna, I can't see you. Okay, this is Tarun. Okay. Yeah, I can see you, right. So thank you so much, and let's work together. Let's join hands, and let's really make fast therapy a reality within this year. Let's not keep it postponed. With if COVID can come so quickly, I think fast therapy can also be started. Okay. Thank you so much, and all the best. Thank you.
Thank you, everybody. Thank you, ma'am.
Thank you. We have our.
Thank you. Namaste.
Just last, I was waiting for this. Yeah, please come here. He's in charge of our SET facility. Please, And he is one person who has been really coordinating.
And myself is Adarsh Kumar Sharma.
Mr. Adarsh Kumar Sharma. And
working as a CTO, Chief Technical Officer here.
yes, thank you so much. And he and his staff has been really helping with all this. Thank you very much and goodbye to everyone. Thank you.
Thank you very much, ma'am. Thank you. Namaste.
Thank you, Gopal. Namaste. Bye.